in Sir Syed College, Shivappa, Kerala, and with the 10 new species and a genus in her credit, Madam contributed a greater proportion to Indian termite taxonomy in the recent times, and she will be delivering a talk on her work.
Good afternoon all. Thank you ma'am for a lovely introduction. So firstly I am saying sorry because most of my slides will be a repetition, maybe a repetition of the former speech. So I will try to stick out my topic, no? So I am dealing with the diversity of termites of Kerala.
But there you cannot see, right?
Take now. Now it's working.
Which one? Which one?
It's pointer. It's moving. It's more.
So we know that phylum Arthropoda is a major dominant or largest phylum. About 84% of the total animal species, described species, were belong to this phylum. Among them, the class Insecta is shows most diverse. About 80% of the total described species were belongs to class Insecta. And they are the key component of our ecosystem because they play an important role in many ecological processes like pollination, major decomposers, and some of them are beneficial insects. Some insects are categorized into a social insect, and they form about 75% of the total insect biomass belongs to the social group. And social group consists of termites, ants, some bees and wasps. And based on the degree of sociality, this social insect is again recategorized into eusocial, semi-social, quasi-social, etc. So we moving to the termites. Termites are eusocial insect, and they are commonly known as white ant, and they are important insect group in our ecological system because they play dual role as a beneficial decomposers. You are familiar with their significance in our ecosystem. They are beneficial insect, and at the same time some of them are harmful group. And they increases our or changing our biotic and abiotic environment, increases our soil fertility, changes our soil structure. So besides this beneficial role, little percent of that is 12.34% of the total termite were comes under the harmful group. So some common men always confused with the identification of termites and ants, but we could identify morphologically, differentiate these ants and what termite based on their what antenna, then their wings, then their petiole, etc. So social organism already discussed as termite colony is considered as a superorganism because in the absence of one caste they could not live. And the colony consists of both animate and inanimate part. Animate part means nymphs, then reproductive forms. Reproductive forms is mainly for the dispersion and reproduction. Then soldier caste for defense. Then worker caste for constructing the nest, feeding, etc. Animate part is the nest and mount. So reproductive caste is that include primary and supplementary reproductives. Then soldier caste, based on their number is varying in the groups. Some groups of termite, the soldier castle is completely absent, especially in the Anoplotermis, etc. And in some groups, very few number of soldier caste is available. And that shows great variation in their morphological variation in the head capsule, mandible, and etc. Here you can see the various varieties of soldiers, and it comes under different families, and their mandible variation and their head capsule variation, pronotum variation. Some of them, their head is prolonged to a nasus, based in the Nasuti termite groups. And based on this pattern, they shows number of individual variations. They show different types of defense mechanisms. Some shows a strong point attack, that is in the case of cryptotermis that have the phragmatic head, so it will help to block the holes. So some are counter attacks, some make labyrinth-type mechanism, some trail guarding mechanism, and the last one is very interesting, autothysis. It is occur in the case of soldierless group, where the worker caste is act as a self-sacrificing. They break down their body and releases some sticky substances that helps to block the enemies like ants, etc. So that is the process autothysis, or it is also known as suicidal altruism. So worker caste is the most encountered individuals in the soldier colony. So based on their, in some groups, Kalashwari Swami sir already mentioned about pseudergates. In the lower termites, there is no true worker caste. Instead of there is pseudogate is present. And they are mainly one-piece nesters because they does not make foraging, etc. So they are pseudo caste and they have the ability to become a reproductive ones. And based on their feeding mechanism, they are different feeding group is there. Group 1 feeders, group 2 feeders, group 3 feeders, group 4 feeders. Group 1 feeders means lower termites mainly comes under this group 1 feeders. And group 3 feeders is the humus feeders, and group 4 is the true soil feeders. Inanimate part means nest that was made up of their saliva, fecus, chewed food items, etc. And this nest may be epigeal or arboreal nest or carton nest, or some of them are mound builders and some of them are open air forages that already discussed about open air forages. And based on this nesting pattern, they are again categorized into one-piece nesters, intermediate nesters, and separate piece nesters. In one-piece nesters, mainly, especially the Calotermitidae group, they feed and nest in the same wood items. Here you can see some of the nesting, different types of nest that I observed during my study. This is the carton nest of Nasuti species. And some of them are found under the rocks or Endotermis species. So generally termites are three types. They are damp wood termites, dry wood termites, and subterranean ones. Dry wood termites means they inhabit in the wood items with very less moisture content that we observed in our furniture like this. And damp wood termites means they live in the wood items with high moisture content. And subterranean means they live in the soil and they make their colonial or foraging tubes on the live or dead wood trees, etc. So most of the 80% of the termites belongs to these subterranean groups. So this also already discussed. They are placed under order Lactodia of cockroaches and Isotrus, now kept in as an infra-order. So you can see the region-wise data. In India, there are about more than 2,940, sorry, not in India, world, extinct species. And in India, there are about 317 species under 54 genera and 6 families. And in Kerala, there is 92 species, 34 genera and 3 families. These are the three families of termites that is observed in the Kerala. Among them, 78% of the overall diversity of Kerala belongs to this family Termitidae. And the second dominant one is the Calotermitidae, and the least one is the Rhinotermitidae. So family Calotermitidae, commonly they are known as drywood termites. Both drywood termites and dampwood termites belongs to this category, you know. And generally, there are key point identification. Already sir mentioned about the difficulty in the identification in termite groups, especially based on the imago caste and worker caste, because they show seasonal variation. That's why the collection of imago caste is very difficult. And worker caste is very encountered groups, but individuals, but we could not get a proper taxonomic key for the identification up to species level in the case of worker caste. So we always focused on the key characters of soldiers. So in this case, family Calotermitidae, the diagnostic characters are their head shape, they are phragmatic, or sometimes they are trach shaped, and their fontanelle region is absent, fontanelle part is absent. Then their tarsal segment is 4-segmented tarsal segment, and pronotum is flat shaped. You can see the diversity in India and Kerala. There are about 8 genera and 51 species in India, and in Kerala there are 4 genera and 12 species. See the generic level: Cryptotermis, Glyptotermis, Neotermis and Prostylectotermis. All these 4 genera were represented from Kerala. See the different groups. Unfortunately, I don't get Prostylectotermis genus. So other 3 genera is there. Cryptotermis, that is obtained in the furniture. Then coming to the second family, Rhinotermitidae, they are subterranean groups. And diagnostic characters here are their fontanelle present. In the case of Calotermitidae, fontanelle is absent, but here it is present, and their tarsal segment is 4-segmented tarsa. Then pronotum is also flat shaped here. Then in India there are 5 subfamilies of Rhinotermitidae were found. They are Coptotermitinae, Heterotermitinae, Pro-Rhinotermitinae, Samotermitinae, then Rhinotermitinae. Of them, the 3 subfamilies occur in the Kerala, that is Coptotermitinae, Heterotermitinae and Pro-Rhinotermitinae. The species composition of these 3 subfamilies are given here. Coptotermitinae is much species, 4 species from Kerala. Then at generic level distribution, India, there are 7 genera and 34 species. And in Kerala, there are 3 genera and 8 species. Compared to India, Heterotermitinae is the most dominant one with 9 species, but taking in Kerala, it is Coptotermitinae with 4 species now. These are the different species or a different genera comes under Rhinotermitidae. First one is the Coptotermis beckeri, second is the Heterotermis indicola, which comes under the Heterotermitinae, and third one is the Pro-Rhinotermis coating, that was a new species, it is accepted. Then coming to the hyperdiverse family, Termitidae, that already I discussed in the previous slide. 78% of the total termites of Kerala belongs to this family. And I think in world, when we take in the world status, 70% of the around 70% of the total termites belongs to this Termitidae family. So they are commonly known as higher termites, and the highly evolved include different groups, that is fungus growing groups, then wood air litter feeders, true soil feeders. The diagnosis of family of Termite is also given here. Their head capsule with well-developed mandible, except in the case of Nasuti termitinae, where it is represented as a very minute spines. But their head is prolonged to a nasus, rostrum or nasus. Then here the pronotum is saddle-shaped. In the other two family, it is flat-shaped. Here it is saddle-shaped. Then the tarsi is four-segmented, except in the genus Endotermis. Actually, this Endotermit is kept as a separate family, Endotermitidae, but recently it is merged with the Termitidae. About 8 subfamilies were found in Termitidae overall, but only 4 subfamilies were found in India. They are Macrotermitinae, Nasuti termitinae, Epicotermitinae, and Termitinae. Here you can see the diversity of termite species in the family Termitidae in India and generic level and species diversity. So much species diversity seen in the subfamily Macrotermitinae with generic diversity, 14 genera, and second dominant one is Nasuti termitinae. Then when we take in the species comparison, it is again Termitinae is much species with much species composition, and overall in India there are 35 genera comes under this family, and the total species is 222 species, that is 70% of total. Then subfamily, when we taking the subfamily Macrotermitinae, Odontotermis, so all of us are familiar with the genus Odontotermis. So Odontotermis is with 42 species, and when we compare in Kerala also, Odontotermis is the dominant 117 species. So total number of species under this subfamily is 62 in India, and in Kerala it is about 21 species. These are the different genera found in Kerala: Odontotermis, Microtermis, and Hypotermis. Then coming to the second subfamily, Epicotermitinae. Here total number of species in India is 26, and in Kerala it is only 5 species. And already I mentioned that in some group is soldier caste is very few number, and sometimes it is completely absent. That was seen in this subfamily Epicotermitina. On this one genus is there, Speculitermis. Only one species have the description of soldier caste, that is Speculitermis sinhalensis. So here 4 genera is Eurytermis, Euhamitermis, Endotermis, and Speculitermis. Of them 3 were as its distribution in Kerala also, that is Euhamitermis, Eurytermis, and Speculitermis. This is, I mentioned, the Speculitermis sinhalensis soldier. So in that case, taxonomic keys for based on the worker caste. So comes to this subfamily Nasuti termitinae. Their total number of genera in India is 11, and with 52 species. And in Kerala it is 8 genera with 15 species. So that genera is Ambulitermis, it is endemic to Kerala, genus Ambulitermis. Then Silonitermellus, actually it was a Sri Lankan endemic genus. Through my study, I could document the Silonitermellus from Kerala. And recently, one of another species also comes under this genus. Then Silonitermis, Emersonitermis, Grellatotermis, Hospitalitermis, Nasutitermis, and Trinovitermis. In the last one, Trinovitermis is a dimorphic one. Soldier is major and minor is there, but in most of the cases there is a soldier major and minor. But very morphologically, we could very easily say that there is a minor and major in the case of this Trinovitermis. So these are the different genera found in India. Then coming to subfamily Termite, the most dominant group when we take in the India and in the Kerala, Termitinae. India with 14 genera and 82 species. Of them, 12 genera has its distribution in Kerala, and species is 31. 12 genera and 31 species. Only the two genera, that is Amitermis and Erinotermis, all 12 is present in Kerala. You can see the difference in morphological difference in this different groups. Anguillotermisus, then all these are categorized in as twisted mandible or a snapping tonsils. They are termed as a snapping tonsils. Of them, the Rhinacapritermis is a new genus that I could document through my study with 2 new species. So total diversity in India, it is about 317 species in different, first 3 families not represented from Kerala. So much diverse, you can see the much diverse families Termitidae with 222 species. In Kerala also it is the most dominant one with 72 species. So total number of species in Kerala is 92 species, and include all these are above discussed slides are not included as new species. It is an unpublished data. Just for by adding this list, it will be increases up to 97. So their molecular work is ongoing. So that why it is not published data. So these are the some new genus and new species that I could collect through my study, and some of them are not listed here. That is Amitermis, that is obtained from the Kavarati, and one Pseudocapritermis species. These are the 2 new species new to India, Dicuspiditermis hudsoni. Both these are endemic to Sri Lanka. New to Kerala. Then when we take into the endemicity, 59% of the total termites of India were endemic to our country. 59%. So on this, when we taking the family-wise data, 71% of the Termitidae is endemic to India. Then second dominant is the Calotermitidae, and the least one is the Stylotermitidae. When we taking the endemicity in Kerala, we have 3 endemic genera, that is Ampullitermis, Indocapritermis, and recently observed Rhinacapritermis, with a remarkable number of species, that is 22 species. That means 24% of the total diversity of Kerala is endemic to Kerala. And when we take in the family-wise data, 73%, 23% of Calotermitidae, and 4% of the Rhinotermitidae. Then already you heard about their ecological and economic significance in most of their former talk. So they play dual role as a beneficial decomposers, and at the same time some of them are reported as a pest species. And some of them has medicinal importance. They are used as due to their antimicrobial activity, especially due to their gut microbiomes. They are used in zootherapy, and they are used edible. They are from the edible group of insect. Then, especially when we take in the pest level, around as per the Rashmi ma'am details, Kerala have about 18 major pest species. Of them, the first 2 belong to the invasive species. We have 3 invasive species in India. Of them, 2 has their distribution from Kerala also. This is only based on my observation during my starting of my study. Actually, it is the colony of, or it is the attack of Cryptotermis dudlei. It was on the beginning of my study work. Very curiously, I just noticed imago cast in my house, in the light of my house, and I just searched about it and find it as and I identify it as a cryptoterm. Then only I could get the data about their sign of infestation. That is the presence of pellets, fecal pellets in the wooden particles. Then only I knew that all my furniture will be completely destroyed by this group. That is, all these are from my house itself. My sofa, attack of the post of staircase sofa, and completely destruct my sofa's furniture. And it is the sign of infestation is the pellets, fecal pellets, and another sign is the kick out holes in the wooden particles. So it is mainly of not this species, mainly of the drywood termites. Then another, it is also encountered in my house, even in the granite, this Heterotermus indicola is obtained, and completely destruct the paper and structural parts of buildings. Another one is the Coptotermis beckeri. Actually, I have not much experience in the practice way, just through the literature, I just say that it is replacing of the damaged wood items. Already mentioned how to damage by using the insecticide and importance to avoid the insecticide application, already mentioned. So I think taxonomic work is not nowadays, it's still actively ongoing, but we need to explore more other things, that is especially already told termitophilous, then barcoding work is, we have the challenge to complete that process, barcoding of Indian termites, then what is ingulins, it's also interesting, then their nesting pattern, their behavioral study, not all this type of work is not occurred in India and not completely, it's most of the cases is not compared to other countries, no, it's very incomplete data from India. Then in addition to this, very significant part of this termite is their gut microbiomes, I think. So not much in India and in Kerala, that type of study is not occurred. So the newcomers and have a chance to start this type of works, because they are the key factor to define which food items that they used based on their gut microbiomes, and this gut microbiome has the property to helps to convert our biological things by decomposition process, act as a bioconversion agents. So need to study more about their profiling of gut microbiomes, and also their isolation of important compounds from them, and their application in their biotechnological and pharmaceutical level. So that's all, but thank you.
Any questions, please? Any questions? If you don't have any questions.
Yes.
In Africa, I got that soldier caste is the last. There is no soldier caste. So how they last?
I'm not saying completely absent, but still some of them have very few number of.
So how will the last caste? What is the reason?
So that is I am telling that we need to explore more and more about all these things, no? Yes, that is your chance, and for further study.
That actually depends on the foraging period. If they go out for long distance, definitely they will have more number of soldiers. If they are soil feeding with the nest and
Yes.
they are short distance foragers, so there is no necessity of having more number of soldiers. They will not divert their energy to develop soldiers, which is actually you can say loss of fitness cost. So that's why the they lost that character, or so probably when they actually adopted that particular environment, so through evolution they have lost those characteristics. So that mainly depends on the length of forest. Yeah, so most of the most of the salicity is usually found at depth of about 10 centimeter, 15 centimeter.
Yes, sir.
So they usually get protected. So usually the ants are the major predators.
Sometimes we could collect the soldier by dumping in about 10 centimeter in depth, no? Just only that we could at least 2 or 3 soldier cast is available. We could spend more time to collect the soldier cast of this group.
Actually, termite soldiers are actually not soldiers. They should come out and defend the colony.
Yes.
So many times we struggle to get these soldiers because when you get hundreds of thousands of workers, there will not be any use for us many times because in the knowing that we cannot identify them. So unless we get at least one soldier, so we feel happy. Identify this. If there are no soldier casts, many times I think no, we struggle this much. So I think that if the soldiers are good enough to defend the colony, they should come forward and they should come and defend the colony so that we will get them.
Yes, kept some of the part for you entomologists to look at. Okay. And we made up Dr. Amina for one question. After now, we still have a problem with your face.
Still,
We still have a termite problem with your face.
yeah.
There's a problem in the house, I say.
So far, we have
Not only in the field, in the house itself, there's a problem.
Okay, I take this opportunity to express my sincere thankful to Kaleshwara Swami sir and the entire organizing team to invite me and to being a part of this program. I'm very happy. Thank you, sir. And thank you, the entire team.
Madam has explained everything. Even identification level, everything she has explained everything. I request Dr. Manjula sir, former Director of Education, to come on.
Our next speaker is Dr. Rashmi. So in fact, when I was at GKVK as research associate under Dr. C.V. sir, so she was working at IWST as a PhD student. So she used to collect termites and come to GKVK for identification. I used to identify termites for her, but later she was actually herself get trained, and later from IWST after her PhD she moved to Institute of Plywood Research and Organization, part of Ministry of Environment and Forest. So there she is working as a scientist. So she will share about the wood preservation techniques.
Thank you, sir. Compared to all of you, my area of research is totally different. All of you see termite as an ecosystem engineers, ketone species. I see them as a pest. Although I worked on the ecosystem services, when I joined my new institute 2017, my director asked me, tell me something about termite. I started. They are the keystone species, the ecosystem engineers. My director told, stop that. We are seeing them as a pest and you have to see them only as a pest. So my institute clearly tells me I have to see them as a pest. And as Amina madam told, 12% of the insect termites are actually wood feeders, and they are problematic to the structures. It's not that the termites are bad to us. When they interfere with our properties, which are economic important to us, then only they are considered as pests. Why we call them as a pest? In India, I don't know, you have the data that how much amount of services they are providing. But you know that in India, the amount of wood produced, out of that, 33% of the wood exclusively goes for replacement of the eaten structures or the damaged structures. And most of them damages comes from the termites. India is a low productive country, forest productivity is very low. So losing that amount of wood just to replace is not a economic or viable option. And 33% of loss is a major loss. So what we have to do? We have to protect the wood. See, whenever we talk that the forests are important. When we are using 33% of the forest product to replace the wood, that means we are using or cutting unnecessarily trees just to replace, not to produce new product. If something is produced out of the tree, that is new product, the value is more. If you are using as a replacement, the value of the product is less. So, to avoid that, we have to protect the wood. And by protecting the wood, you can protect 12% of the, you can reduce the cutting down of the forest up to 12%. See, what do you mean by protection? Protection means preventing it from the insect or the other kind of attack. Usually we talk that the forest chemicals are bad, but chemicals are not bad. They are protecting your wood. Indirectly, they are also protecting your forest. There are different methods of forest protection. Chemicals are one of them. If you are using very harmful chemicals to protect the wood, then it is bad. If you are using the chemicals which are not that highly damaging to the environmental health, then it is acceptable. Always whenever you see, there is always a line where it was given. How much you can use, how much you should use. If you are using the chemicals to protect the crops also, nobody tells that don't use the chemicals to protect the crops, because it gives the food and in turns it survives most of the human society. Similarly, protection of the wood is also important, and protection means enhancing the durability of wood with the help of chemicals, so that we can lengthen the service life of the wood, protect it from the fungi and insects, and guard against the weathering. When we talk about the wood degradation, there are two types. One, abiotic, another biotic. Biotic because of your living organism. Abiotic because of climate. Like UV rays, water, rain, whatever those things are there, no, those also affect the wood. You have to use protection to prevent the wood loss. So you have to use preservative. What kind of preservative you should use? A preservative should be highly effective. It could kill different kind of animals, the broad spectrum. It should be easy to handle, and it should be easily available. If it is a very costly, like we are talking about the pheromones and all, they are not easy to handle, not easy to, not pocket friendly. They are very costly. That when any material or any product which you produce becomes costly, the person or the end user never want to use that product. So I was asked to deal about the wood preservative. In your agriculture sector, you use that different kind of polished terms like, okay, this kind of pheromone kind of, botanical kind of, and all those preservative. In wood industry, it's a kind of crude industry. It uses only 3 types of preservative mainly: tar oil, organic solvent, and the waterborne. Tar oil means it's nothing but a bituminous oil. You might have seen that black color oil. Most of your old houses, if you see, made up of wooden, they will have that black coating. That is nothing but the tar oil. That tar oil is very long lasting, effective against the all kind of fungus and the insect, but it is not used at every point, because the tar oil gives the black coloration. That result in the reduction of the aesthetic value of your wooden material. And after the usage of the tar oil, it is not possible to paint it over. So, then there are waterborne chemicals. Waterborne chemicals means these are the salts which are dissolved in water. There are 2 types of waterborne chemicals: leachable and fixative. Leachable means with the water or the rain which will come out of the wood. And fixed means it won't come out even after the contact with the water. See, what is the benefit of waterborne chemicals? They are colorless, odorless, it can be painted over, but the problem is it has to be dyed properly. The wood is an organic material which has the ability to absorb the water and give out the water. While painting, if you put more water and if you dry directly on the sunlight, there is a chance that wood will get wrapping, means folding, or the cracks will break. To avoid that, you have to dry it in the shade or it will be cool dried. Then we have organic solvent. The wood preservative which are mixed with the spirit, just like your paint. The paint you will mix with the kerosene or other kind of some white color spirit, they will give Similarly, this preservative has to be mixed with the spirits and they have to apply it. These are the very useful. They are easily paintable and they can be used in the indoors. But they have smell because of the spirit, and because of that kerosene or the spirit nature, it is easily flammable. So these are the three types of materials we are using. But all these chemicals are not accepted everywhere. Because of the health hazard, there are many problems. For example, you have CCA, copper chrome arsenate. The copper chrome arsenate is the wood preservative produced in the Forest Research Institute of India in the year 1938. And so far, none of the chemicals or the wood preservative are on par with the CCA. But we are not allowed to use the CCA considering the presence of the arsenic in that. So, CCA is not banned. Many people have the misconception that it is banned. No, it is not completely banned. Only it has a restricted use. It can be used in the areas where direct human contact humans won't come in contact with the CCA. So some of these chemicals are still in use. Botanicals are also tried, but they are not very effective to give the required amount of protection to the plant. See, I was also asked to cover the methods of application. Whenever timber is used, there are some methods we have to follow. Why we have to follow? Because we are given with the BIS rules. Bureau of Indian Standard, IS 401 clearly talks any timber which you are using has to be treated, and that has to be treated in the particular manner given in BIS 401. One is a brush and spray, then the immersion and the diffusion. Brush and spray is the common technique where you are brushing the oil or the preservative. Then diffusion means you are putting the materials in the chemical. Then we have hot and cold process as well as the bruchery process. I won't go in these processes detail, but I just want to talk about wood restoration mode. What is the difference between wood preservation and the wood restoration? Wood preservation means you are treating the wood initially and producing the material out of it, or any wood which is used is that you are treating with the preservative. The restoration is totally different fact. Restoration means you have a wood which is already used, which is of historic importance, and you don't want to lose that. Here, when we talk about the restoration, it's not about the economic value. It's about its historic value, all the histories it carries with itself. The preservation is of economic importance, whereas the restoration is of historic importance. See, we got some these beautiful structures, no? Wooden artifacts. These are the structures that are in the Lal Bagh. These structures as if don't have huge history. The upper four structures you can see, no? These are in the Lal Bagh, and the lower four are in the Kaban Park. What happened is these are the structures prepared from the trees which are there or planted by Hyder Ali and Tipu Sultan in around 200 or 250 years back. Those trees planted by the Hyder Ali and Tipu Sultan fell down in the year 2017 due to heavy rain. So what happened is, usually what is the procedure when the wood fell down on the forest department or any of the horticulture department. They usually call for the auction. They sell it to any of the timber yards. But these having the historic importance, they didn't wanted to sell that. So they called the architects throughout the India, even the architects from the Shantiniketan of West Bengal, Rabindranath Tagore Shantiniketan, they came and carved the different structures, beautiful structures, and those structures were implanted or kept in the display in the Lal Bagh and the Kabban Park area. There are totally 70 structures. Out of that, 20 already gone. When we got this work, there were 50 wooden sculptures which they asked us to restore or preserve. And this was a new work for us. We usually deal with the wood which need to be preserved or before use. Here we had the wood which was already produced into something which cannot be disturbed, and we have to decide how we have to treat them. So our modus operandi was to first inspect the artifact, to see what kind of infestation was there. Then how to design the experiment based on design the treatment protocol based on the wood structure. When we inspected, we saw that termite attack was there on most of the structures. Fungal attack was there, carpenter bee attack was there, pinhole borer attack was there. These were the insects which in fact affect the wood. Other than that, when we talk about the abiotic degradation, these sculptures were kept in the open. So there was a rain falling on that, dust falling on that, over that sunlight was there. Because of that sunlight and the rain, there was an end crack, split, and the discoloration of the artifact was also seen on these sculptures. See, what exactly now we have to do is we have to treat where the splits, discoloration and cracks are there. So we have to go for the primary processing. See, one thing you need to always understand that these are not the smaller structures. You have seen that Buddha structure. That is around 18 or 20 feet tall Buddha structure it is. That wisdom teeth next to Buddha, that is also around that much, 20 feet tall. All the structures were more than 8 or 10 feet tall. They were already placed at one point. They are already carved. You cannot simply clean them. If you want to dust them, you cannot simply dust them because there are minute carvings. You have to be very cautious, very clear, and you should design what after what you want to do. So the first thing was dusting. We have to dust them, then wet cleaning. The wet cleaning means usually you do it with the water, but here we poured lots of water. All the wood will absorb the water. Here in these structures, what happened is lots of insects were inhibiting. Those were not actually wood-eating organisms, but when the wood holes or the cracks formed, many insects started staying there. So the first step was to remove the insect out of that. So we gave the wood treatment or the water treatment. See, you can see these pictures, no? You just see this picture and tell me what is the problem with this. Any guesses? Okay, I'll go to the next picture. The same pictures. Now you tell me the difference. Oh no, discoloration is afterwards. This is after treatment. Here, the first thing is wood is kept directly on the contact with the soil. Soil. Whenever we are talking about the termites are the soil organism. A small point of entry is more than enough to destroy the whole structure. So the basic or the thumb rule you can tell is whenever we are using the wood for any of the components, structural or any of the component, they shouldn't be directly in contact with the soil. They should be always separated from the soil, or if they have to come in contact with the soil, the soil which you are using should be properly treated. So the first thing was we were supposed to treat the soil. So soil treatment can be done by two methods, by creating the chemical barrier or by creating the physical barrier. Chemical barrier was created by any of the chemicals which can be act as a insect repellent. So we used any insect repellent, did the soil treatment first, and to create the physical barrier there are many options, just like creating the mesh and all. All those were not practical for us. So we did the simple concrete structures we prepared, and over that concrete structure we asked them to keep those structures. The first thing is we were able to avoid the termite attack because of this simple technique. We always remember that if we follow the simple techniques and the simple rules prescribed, we don't get most of the damages occur to any of our structures, let it be housing structures or any of the artifacts. The next was boric acid and the borax treatment. See, while starting the PPT, I told about 3 types of treatments: oil treatment, waterborne treatment, and the solvent treatment. So we were did the first thing was we did that soil treatment. But if anything comes in contact with the ground, another treatment is possible is tar oil, application of the tar oil. But while telling the tar oil, I also told that after the application of the tar oil, you cannot provide varnishes, and it interfere with the beauty of your artifact. So that was avoided. The next we went for borax acid and boric treatment. That is the most safest treatment which was given for any of the wooden artifacts or any of the wooden material which we are using in the house, like any bamboo kind of material if you are using to prepare a toy and all. What is specified treatment is boric acid and borax treatment, because it is not at all harmful. Like if you have the toys, kids will have the habit of putting all the toys in the mouth. It shouldn't be harmful for them. So to avoid all that, we give boric acid, borax treatment. The artifacts has lots of insects, and it was kept in open. Even after the treatment, again and again insects were coming. So to avoid that, we have to treat with boric acid and borax. Boric acid and borax act as it's a water soluble, leachable type of treatment, but it avoids the attack of the insects. To avoid that attack, we did the boric acid and borax treatment. But here, after boric acid and borax treatment, if water or rain comes, it will leach out. So until we go for this treatment was done, so that until we go for the next treatment, it should prevent the entry of the insect. So next we treated with the CCB. In the waterborne treatment, there are 2 types: water soluble, that is leachable, and non-leachable. Fixative type. CCB is the fixative type of treatment in which copper sulfate acts as a fungicide. Chromium is a fixative agent, and boric and borax work as a insecticide. After that, we treated this whole artifact with the CCB. Here the treatment we know that CCB we are doing, but how to do that treatment was a major trick, because the artifacts are of different shapes. Whenever we treat the wood, there are 2 things we have to remember: retention of the chemical and the penetration of the chemical. What is penetration of the chemical? You have a wood which is a solid material. If you are simply spraying the wood, the whole chemical won't go inside, and the through and through treatment of the wood won't happen. So that you have to keep that wood immersed or for a longer duration in the preservative. With the this already carved structures and the long structure, it was not possible to take a container and dip it. So how you have to do? So what we did was we took the gunny bags. We tied the whole structure, 6 feet tall and 6 feet long, more than 6 feet long structures. We tied them manually with the gunny bags and sprayed the CCB over that. And this we repeated for every 2 to 3 hours and 2 to 3 times. Whenever the gunny bags get dried, you have to again spray the chemicals and you have to wait, so that your chemical has to penetrate inside the wood. See, and another major study, if you see these structures, no, I was talking about the cracks and the carpenter bee attack. You just see how much big cracks have happened. The whole thing has broken. And this was the fifth structure where the whole fin is totally broken. You have to repair it and restore it. There you see that is the carpenter bee attack. So how you have to do? You have to fill that first. How you have to fill? Whenever you fill anything, you cannot simply fill with any kind of the wood. You have to see what kind of the wood is used for that particular matter. That material is of, and you have to select the wood or the wood of same species. You have to chip that, make into small pieces, then separate the those wood chips into smaller particles. The particles will be of coarse particles and the softer particles. The coarse particle will take more resins. And you cannot use the coarse particle on the outermost layer. So you have to mix the coarse particle with the resin. Then you have to fill that. Again you have to take another round of softer particles. If needed, coconut shell chips, all those things you have to take, mix with the resin and fill it. After filling also you cannot do the filling at one sitting. Filling you have to repeat. You have to fill half the portion. Then you have to see how much it is swelling. After that filling, how much swelling is happening. So you have to wait and then decide how much you are going to fill. Like that we separated the particles, then mixed with the resins. Can you play that video? Here this is the carpenter bee attack. And you just see the height of the structure, okay? That video is enough. You see the structure. It is taller than the boy standing next to that. You have to fill that whole structure. And when we were working, no, we could even see. In that, there was a carpenter bee actually working on that. It was staying in that. He is trying to remove that carpenter bee, but it is not coming out. So then finally we, along with the carpenter bee, we filled the. So there were the 3-4 carpenter bee size, like your whole finger will go inside that. That much big. Another video is also there. It was there and you have to fill that. In that case, these are the smaller structures. We fill them mixed with the fevicol. So you just see how much that inside it is going, that nail. That rod is there, no? How much inside it is going. That much inner we have to fill the particles. So those big holes filling is it looks simple, but that time we didn't know what to do. So we were not sure what particle size we should use. So somehow it was like a trial and error. We tried and we somehow filled that. And here you can see, there are some cuts are there. Those are the cuts because of the weathering. Because of the sunlight, what happens is, when sunlight falls on the wood expands. And after that, when the winter comes, again that swelling and shrinkage will happen always in the wood. With the water also same thing happens. And when the water fills inside and when water has to go out, it breaks the structures. Especially with these woods, which are artifacts made up of the wood which is 250 years old. Just imagine, all the stability and the mechanical properties of the woods are lost. They were supposed to treat before the preparation of the artifact. They didn't do that. So it was okay, we are moving towards a dead end. Just we have to save that. Other than that, wood will have other kind of defects, such as fungal attacks. Some things will eat on that because of the falling of some materials or rain. The chipping out of the things will be there. And whenever this chipping out of the things will be there, there is an opening for the fungal attack. So you have to avoid that. Here comes our creosote oil treatment or the tar oil, which was told first. The ends were coated. After that, there were small holes made up of pinhole borers that you cannot treat with any kind of chemicals. So you have to manually inject the insecticide. Just like that, you have to manually inject the insecticide inside the hole. And the other things you have to fill with the insecticidal dust along with the fillers. Fillers means nothing but sawdust particles or the coconut shell particles. That you have to mix with the insecticide, mix with the resin depending upon what kind of resin you want, what kind of finishing you want. Accordingly, you want to marine grade plywood or the epoxy resin or the melamine resin, what you want, that you have to decide, and accordingly you have to mix with the resin and you have to apply. After application, you have to give the shape which was there initially. So after that, we are using oil treatment. We did the water treatment both leachable and fixed. We did the tar oil treatment. Now we are left with water treatment. Why water treatment is given when we have already given the CCB? Any idea? No, what is the relationship with the water and the oil? Oil repels the entry of the water. It acts as a protective cover. And many of these oils like neem oil and the currant oil have the insecticidal properties. So whenever the cracks is there, if you fill the oil with that, it won't, it repel the entry of the, it prevent the entry, direct absorption of the water inside that wood materials. To avoid that, we are using the water treatment, oil treatment. So we use neem oil and the currant oil. You can remember that artifact which is there. I have showed you where the biggest crack was there. After filling, that was the final structure. We filled all the gaps, we filled all the cracks with the wax, and then we treated with the CCB, and then we treated with the oil. See, you see the height of the structure? Just imagine what would have been the challenge for that. And why? This was most important because oil treatment, this was kept in the garden. Garden is the area where always there will be a presence of water and it will be always moist. So the chance of attack will be always more. So to avoid that, we had the oil treatment. After that, then the varnish treatment. This is how it looks before and after. See how many cracks were there in this mother family structure. This lowermost structure is there, no? That is called as family structure. The one was before treatment, one is after treatment. And this is the fish which was in a very bad shape. If you see that first picture, the fin of the fish was totally removed and kept aside. And whole inside hole was carved by the insect and there was lots of insects staying in that. So we have to fill that, fix the fin, and final product was like this. So it was brought back to the earlier structure. In conclusion, we can tell that artifacts treatment based upon the type of the wood which was used for the preparation of the artifact itself, then depending upon the condition where the artifact was placed, depending upon that you have to decide the treatment protocol. And the wood treatment is not once. If it is kept in the open air, you have to always monitor the artifacts or the wood material for the treatment. This is one kind of that. But if you have a difficult situation like this, where one portion is fallen off and you don't know how to totally repair it. So here we did the same thing, cleaning. Here you can see the cracks now, how big cracks were there. If you allow that cat to remain like that, over the period of 2 or 3 years, it completely broke and fall off. So you have to fasten that with the help of screw. Then you have to fill that with the fillers, then treat it. And here we use 2 things, coconut shell powder and wood particles along with the marine water grade Fevicol. So we filled all those inner structures with the porous particle. To give the smooth finish, we filled it with the coconut shell mixed with the Fevicol. Then we don't know how it looked initially. We don't know. Only thing we know is it was the bird statue, bird statue. So we have to take the measurement. You have to calculate based upon its body size what will be its beak. Then you have to prepare the beak by giving the reinforcement. Reinforcement means you have to give some structure so that it can hold and remain for the longer time. Here we gave the reinforcement initially with the iron nails. So we decided the amount of the beak, how long it should be, where the depression should go. So accordingly we placed the nail. Then manually we prepared the beak. But only with the iron rods you are not supposed to, you can't give the proper reinforcement. So what we use, that white color, it's the glass mesh and the glass fibers. Those two things, glass fibers and the glass mesh, are used to give the reinforcement in any kind of plywood or the panel wood structures. So we use the glass mesh, we gave the another level of reinforcement so that the beak should be holding. Over that manually we prepared the beak, then we gave the final touch to that beak. Then we have next stage piece, we have to varnish it. After varnish, this is how it looked, before and the after. So however, we tell now wood treatment is easier, but there are lots of things involved. I just took an example of only wood artifacts. Then you have the structural timbers, plywood and panel woods. Different kind of materials are there, and depending with the material, your treatment, and depending upon your end users, treatment protocols and the duration will vary. Thank you. Any doubts? Yes.
How will you decide which treatment we should go?
See, we have to see where it was placed. See, that's why I took this initially, what kind of preservative we have to use. You know that the artifacts were kept in the outside. So you need something which will prevent the water absorption for the longer time. Then you know that the insects were attacking that. You need something which will prevent the insect attack. You know that fungus was attacking. You need something which will prevent the fungus entry also. So that's why we went for CCP. So you decide what kind of insects are attacking your artifact and what can be the future attack. Like you have to see the seasons, how the weathering will change, and what might be the other appearing organisms according to season. According to that you have to decide. When it comes to wood, there is one always saying, always before using the wood you just decide what is the end use and what is the product you are going to, where you are using it. That is the main thumb rule we tell. Whenever we are using the wood, for that only, if any wood comes into the India, we have to test that in Indian condition. That also not at one position, 6 locations, because India is a big country with different climatic conditions. We have to test that, then only approval comes, if it stays in that particular area. The wood preservation is always species specific, location specific.
Is there any case studies related to the household wood management using these?
Yes, there are. Because if you see the Hyderabad, there is one institute which exclusively related to this structural wood treatment.
What is the endurance then? Lifespan of these protection agents?
Protection?
How long it will give the protection?
That is also depend. But usually these chlorophyllipos are used to give the long protection.
No, I'm speaking about this one. You said organs,
Okay, wood preservative. See, these will give the 2 years or 3 years. You have to repeat the treatment. That's why I told long-term monitoring is always needed. You cannot tell that, okay, you gave the treatment and okay, it's final. For the lifetime it is, okay, you cannot tell that. You have to repeat the treatment if needed. You have to observe it, whether any insect is again attacking.
So, what about the economics then? I mean, if you go for commercial.
See, I told there are some things which are of historic importance, not the economic importance. These are made. They could have simply sold that. They would have got the money and used the money. But they don't want it to sell because there is some historic importance attached to it. And you cannot compare the historic importance with the economic importance. If it is only of economic importance, rather than treating, if it is feasible, go for the replacement or then take a new structure. For example, we had one consultation, one house. She spent 25 lakhs for the interiors, only interiors. And within a 2 years, she got the attack. Even she tried many inside. She got this powder post beetle attack. We told her, whatever you do, this won't last. For the best treatment method for the powder post beetle is fumigation. And fumigation you cannot use when the people are staying in the house. Even if you close the house and inside the house, you cannot give the fumigation. If you have the small structure, keep it in a chamber and give the fumigation, that will work. For the house which was attached, house furnitures or the interiors which is already attached to the walls, you cannot give the fumigation service. So in that case, we tell them to replace it so that you can save the other wood which is not attacked. That is the only solution. That time it comes the preservation, not the restoration. Restoration means you are bringing back that structure back to its earlier state. Yes.
Regarding pillars, you told that we need to use the same species as the same species. If species is not available, as I mentioned about Nagbar and
See, the species were very easily available: mango, eucalyptus, and the mahogany species. That is highly available in that. If it is different species, sometimes what happens is they may not mix properly. That's the only thing. There will be available, otherwise you can try with some resins will provide, you can change. See what happens is density of the wood changes with the age also. So what exactly comes in question is the density. If you are using the lower density of different wood, which will make in correlation with that particular wood which was used, that will also do. But usually we take care. Like if you are using the teak wood and you use the filler of poplar or the rubber wood, that means that rather than the earlier attack, the more attack will be there because of the rubber wood. So to avoid that. Any other?
Remote talks are left out. So we will discuss with her after session also. Thank you everyone for giving me the opportunity. Very beautifully about preservation and restoration of food and experiment with wooden artifacts. But really it is very practical, and the persons who will build the house and all, we should be very careful how we can protect our interior everything. Okay, and further discussion you can have with her after. Anyway, Madam, on behalf of our Department of Entomology and University and Extend, thanks for your beautiful speech. Now you get
The next speaker is Dr. INR sir, and in fact we collaborated with one of the works. When I got the specimens from Deputy Director of DPP QS, Dr. Nagaraj, and I could able to see that it was actually a Coptoderma species, but it was not reported from India. So then INR sir was actually collecting specimens from Tuticorin, the place where the usually the imported wood reaches, especially only I think Vizag, and the Tuticorin actually receives the imported wood. And when the imported wood contains termites, means it was actually it's of great importance. So I think that I was telling in my talk. So sir is actually involved with the quarantine system for about 35 years. So he'll be sharing his experience of a quarantine system at the same time about termites as well. So thank you, sir.
Good afternoon to everybody. I am very thankful Dr. Kaleswari Swami sir and Dr. D.K. Nagaraj sir, Deputy Director of our DBPS. minor PPO retired. Our DPPS is under the is the subordinate office of Ministry of Agriculture. Our secretary in our ministry, secretary, then additional secretary, joint secretary, then director PP. Joint secretary will look after in charge of DPPS, Director of Plant Protection, Quarantine and Storage, National Institute of Plant Health Management and Monitoring of Pesticide Residue at national level, Delhi. Then Director PP will look after IPM unit, PP1, PP2 insecticide unit and PPS on the plant quarantine. Then under this Director of PPQS, PPA is our head of the department. Under PPA, 4 APPA and 5 APPAs are there. 1 APPA IPM, Integrated Pest Management Center, then another APPA plant quarantine, then another, now not JD, PPA is there, APPA is there, locust control, then another APPA CIL, Central Insecticide Laboratory, and another APPA CIBRC, Central Insecticide Board Registration Committee. Under APPA IPM, regional IPMs, 6 regional IPM stations are there. In that, under that, 30 sub-officers are there. Like that, under APPA PQ, regional PQs are 7. 7 officers are there. And the plant quarantine stations are around 86. Total 92 stations are there. Then LW, Locust Warning Organization, field station station investigation of locust, 10 stations are there total. Then APPA CAL, he will look after insecticide laboratory and the regional pesticide testing laboratory. APPA is the Secretariat of CABRC, and licensing issue for insecticide company, licensing issue authority is APPA CABRC. Then our biosecurity, our Indian biosecurity, these are the ICR institutes are involved to look after our biosecurity of our nation. Actually, total 92 plant quarantine station. Out of that, 7 regional plant quarantine station. One is at Bangalore airport also, one RPKC is there. Then 36 IPM centers, 12 locust warning organization, 1 central insecticide laboratory, 1 regional pesticide testing laboratory, and 1 national institute of plant health management at Hyderabad. Then these are the important regulations. Destructive Insects and Pests Act 1914. This is the first act come to India to protect the plants, plant products, and animal, plant and plant products for the new entry of insects, virus, and pathogens into our country. And also domestic quarantine also, from one state to another state, it will control. Then after that, then PFS order 1989 came. Plants, fruits, seeds order 1989, regulation of import into India. As per that order, they have to get phytosanitary certificate from the exporting country, and they have to submit in our country. Then they have to apply permit also. 1 month in advance, what are the commodity they want to import, they have to apply permit, consent, RPKs. Then this the Insecticide Act 1968, it is an act to monitor the import, distribution, and transportation, and usage, and what it will not, and to look after any harmful for insect human beings and animal beings. That is the insecticide of 1968. India is our NPPO is our Indian Directorate of Plant Protection, Quarantine and Storage, situated at Faridabad, Haryana State. And our NPPO contact point is Joint Secretary PP, Ministry of Agriculture. Then this first Destructive Insect and Pest Act 1914, then PFS order, then Plant Quarantine Order 2003. This order came, and then PQ, Plant Quarantine. This what are Plant Quarantine Act, and this is a biosecurity Plant Quarantine Order is a protect our biosecurity. That is any quarantine pest or whatever that pest not present in India. It is an act to prevent the plant quarantine pest enter into the India. Then introduction is spread of exotic pests that are harmful to crops. That is also exotic pest means quarantine pest. That is quarantine pest means that is potential economic importance that is not present in India, or if it is present, it is not widely present particular area, and it will be controlled by official. Then this monitor of import agriculture committee as per agreed condition. Import whatever commodities imported, it should come through the concerned airport, seaport, or land frontier, or ICD, or post offices. That will be mentioned in the PQ order 3. If it is not come through that entries, it should that relaxation can be granted only by ministry. First, gain market access for India's agriculture. That our Indian importer can assess the market, what are the commodities, where it is cheap, everything they will first analyze. Then they will submit a PRA application, because if it is not present in the PQ order 2003 new commodity, they have to apply for PRA, pest risk analysis. Then after completion of PRA, that draft notification will be issued in the ministerial website. After 6 months, that gazette notification will come, then it will be included in the PQ order, and importer can easily. These are the
basic students will knowing this. Our
only few. Termites, only few. This NSPM 9 is concerned with this pallet, wooden pallets. Wooden pallets going to export should be fumigated with MBR, methyl bromide, 40 grams per cubic meter for 24 hours. And they should mark in that pallet. The company, the unique mark will be there. They have to mark on the pallet. NSPM 11, it is quarantine treatment of with methyl bromide. Methyl bromide is in gas which is greater than air. So in our methyl bromide, 90% methyl bromide content and 2% chloropicrin. That chloropicrin will is acting as a warning agent, because methyl bromide there is no smell, nothing. Only this. Stages market. First in import, first they have to see the market, which are the country commodity available. Then bilateral agreement. Then pest risk analysis complete. Then potential quarantine pest. What are the pests available? Then system approach. Then scheduling in peak order. Then phytosan certificate. If anything required. Then inspection on arrival. After arrival of the consumer, peak officials will inspect the cargo. Then if necessary, treatment will be carried out. Then release the cargo. This is the peak order 2003. Schedule 1. These are Schedule 1 entry points. Entry total 47 seaport and 25 airport and 24 land frontiers, and 11 international post office. By that they can import. Schedule 2 inland container depots. Inland container depots. By that PQ officer will go and inspect the cargo and release the this one. List of foreign post office, that 11 post offices are there. Schedule 4 list of plant or planting material countries from where import is propagated. These plants and plant material they cannot import it. What is the reason? That is schedule they will mention. Schedule 5 list of plant and plant material imports which are restricted permission. These materials can be imported only through some institutes. Others they cannot import it. Schedule 6 list of plant and plant material permitted import with additional declaration and special conditions. Schedule 6 all timber logs should be fumigated with MBR methyl bromide 40 grams per cubic meter 24 hours. Like the 7th schedule. 7th schedule no additional declaration, nothing. But whatever 7th schedule, some logs also there, wooden logs. Whatever logs they have to do fumigation, and after if any pest is there or not, we have to do compulsory fumigation and release the cargo. Like that cotton also. Cotton, because cotton, so many 3 quarantine pests are there, foreign countries, USA, Australia, and Endemism grandis, Endemism vestilis, and Endemism peninsularis. So it should not enter into India. That's why we are precautionary. We are doing compulsory fumigation. After reaching India, 24 grams per cubic meter, we are doing cotton fumigation. Schedule 8 list of quarantine weed seeds. If any weed seeds imported along with the consignment, it should be deported. Only deported, no treatment and nothing. Because already 2 to green we got so many quarantine weed seeds imported, and we did around 500 containers we deported the cargo. Schedule 9 inspection fees, fumigation fees inspection fees detail. Schedule 10 list of permit issuing authority. Who are the permit issuing authorities? It is mentioned. All state, all agriculture department director they can issue, and they can authorize to issue JD officer, JD level officer also. Then PQ officer they can issue. Schedule 11th list of inspection authority, post-entry quarantine. Suppose any plants, seed or propagation material will come, it should go for post-entry quarantine. First they will conduct a designated inspection authority, then they will visit the place, and if it is suitable, then they will recommend. Then they have to apply for import. Then on arrival of the cargo, PQ officials inspect the cargo, and they will send to the post-entry quarantine facility. Then if any symptoms of virus or fungus or any pathogens will observe, the consignment may be destroyed or treated. Treatment may be required. Schedule 12th quantities. What are the how much quantity can be imported for NBBGR? This post-entry quarantine planting. This is the SOP for post-entry quarantine. Already informed. After reaching the consignment, they have to keep in glasses or polyos or isolated field. Then this designated inspection authority and PKS officer will go and visit a place in particular interval. Then if no symptoms after the completion of period, they will release the cargo. If any symptoms develop, they may destroy or treatment may be recommended. Inspection consumption material. How to collect the sample from first primary sample we have to collect it, then composite sample, then labeling of sample, preparation of submitted sample going to lab, and sealing and observation under microscope. Any pest, anything is there, we will recommend the fumigation, and after that release if any pest and disease. Pest inspection of material container. This is wood container. So pests are there. So we have to collect inside. Suppose the timber logs pest always present on the above the bark and below the bark also. We can see the symptoms. Some powdery form or that sawdust, something will be there. So that means inside some insects will be there. So we can remove the bark and we can found insect. Suppose any pinholes and pinhole short coats are there, we are using this chisel and a hammer, and if we are collecting insect, and after that we are recommending for fumigation. In vessels, more or less important. These are the vessel inspection. Suppose we are going for vessel inspection. Suppose any insect we observe, then we will recommend for fumigation, vessel fumigation. Then we will ask the captain of the vessel how much volume each hatch. Then accordingly we will go and give the dosage for 24 hours, 48 grams per cubic meter, 24 hours. Then crews may go outside the ship. Only captain will be there inside the vessel. So he will look after them. Then after completion of 24 hours and degassing, crews will come and enter the vessel. These are Dr. Nagaraj sir and myself, inspector. Dr. Nagaraj sir did a very great effort and very good things for Tuticorin office. Earlier Tuticorin office is very last interception. But when SAR came, so many quarantine pests, so many quarantine beetles we collected and reported, so many non-complaints reported. Due to all, go to Dr. Nagaraj sir effort. This is the interception of Gurbs species, Gurbs larva in pine logs imported from USA. Gurbs is a quarantine pest, USA. And this also, Gurbs larva, pine logs. Generally it will come. You are doing fumigation. And another one more pest, Cerambycidae adult, Monocomus carolinensis also we noted and did fumigation. Populus, this is imported from Belgium and Germany. This also we are intercepted Paratora laticolis insect, that is quarantine pest, and we recommended so many consumers because there we are MBR based out country. They cannot, they are not doing fumigation. So after reaching here, we are doing fumigation and releasing cargo after charging 5-ton inspection fee. If the consummate is fumigated there and coming here means we will charge only single time. If not fumigated there and coming here and we are doing fumigation, 5-ton inspection fees we are charging. These are Paratora laticolis and this in Belgium and Germany. These are Belgium. These are the consent 2018, 19, 19, 20. These are the consent intercepted. These are the non-comings we send documentation. Documentation to sometimes fake PSC also they are issuing. Fake, they are submitting fake PSC. So if fake PSC means we will deport the cargo. If they request, they can request to JS, PP, Ministry of Agriculture. If after verification document, sometimes he may ask to release the cargo after charging 5 times. If the consignment is free from pest and diseases. These are the total non-compliances, 2231350, total 2784. Non-compliance, when we are intercepted insect, we are sending non-compliance to the countries, particular country. Import agriculture commodity. Fungi, these are the pests intercepted during 2017 to 19. Bacteria, fungi, virus, nematode, insects, weeds. International Standard for Phytosanitary, ISTM 15, that is International Standard Phytosanitary Measures. It is governed by IPPC, International Plant Protection Convention, which is under the control of FAO. That is FAO consists of 195 countries and IPPC consists of 183 countries. These are the closely of phytosanitary terms, ISTM 5. ISBN 7 export certificate system, ISBN 12 guidelines for 5000 certificates, ISBN 20 guidelines for 5000 import regulatory system, 23 guidelines for inspection, methodology for sampling, ISBN 31. National, this is the NSBN 23 newly introduced by our government. So far, only government officers can inspect the export consignment. Now they are given private. Now the private people will inspect the cargo and report online report. They will submit the officer in charge of plant quarantine system. Then after checking everything, he will issue the certificate. Interesting in Suriname, Beltogeny loss. This is the Coptotermus testis. Yes, this insect intercepted from Tutugurin plant quarantine office and sent to Dr. Taisuke sir. We are very thankful to sir. Sir identified and we sent a non-province to that country. This is video how? Video. So many termites we collected. It is below the bark we collected. Huh? Coptotermus testation. This paper is published by Dr. Taisuke Kanoa sir, Dr. Nayaka sir, and we sent identification. So we got two species of genus Coptotermus intercepted. That is widespread among the. That is imported through Yemeni lox, Moro, and Tally lox imported from Colombia, Suriname, and Equatorial Guinea. These two indigenous species are non-native to India. It is not native. It is invasive pest. So it will accommodate in the changed environment condition. Identification was initially done by what sir told, morphological and DNA barcoding. So single blade native of America. Both the species are the serious pest the pest interesting. So we have to infect thoroughly. Even any pinhole, anything is there. We scarab beetle, this one, and Ceramphisidae larva. Tracing video. Ceramphisidae larva imported from Suriname. So many larvae we collected in our office. is Cardilomera spinicornis. This is Africa, imported from Africa. This is also quarantine pest. This is Capratum testis. This is another quarantine pest, termite. This is Cardilomera spinicornis. This one. I collected in the container. So much. That time around 50 specimens I collected. Native of America, Africa being reported from many countries, Angola, Benin and Cameroon, etc. Erythrium, this is Tollylax, mainly it is interested from Tollylax. The interest on these logs is not, it is not present in India, imported from Cameroon. This is Ceramphisidae larva in USA consumed. Yes. It's a typographus. That is quarantine test. These are larvae inside a larva. So much larva. Is it's a calligraphis. This is Paratora lacticum, imported from Belgium and Germany. This is Siloricus pest. In poplar python. This is Pyrotail larva. This is video. Gurbs larva, interested in Cameron Tallylux. This also. Ceramphisidae, Cameron Tallylux. This is Monocomus carolinensis. Monocomus carolinensis. This is intercepted from the consignment of Colombia and USA. This is a powder post beetle. We have to do fumigation. That is quarantine imported from animal dry Niger. Okay. This is inspection, ship inspection. This they want Australians. They want free from ash and gypsy moth. That particular vessel. Particular vessel is carrying that solar fans. That's why we visited all whole vessel and find any Asian gypsy moth. There is no. After that we are issued certificates. All stores, everything gone. Thanks.
This methyl bromide treatment, no?
Yes, madam.
It is
Ether,
All the countries are not accepting the material treated with the methyl bromide, sir.
madam, our country requirement is only MBR. No aluminum phosphate, nothing. No.
Then in case of we are importing. If we are exporting.
Exporting, we are doing as per importing country requirement. As per importing, because aluminum phosphate will not kill egg stage. Only it will kill adult and larva. Egg stage will be remain in active stage. Yeah, the ship may go after 2 months concern port. In the meantime, egg will develop into adult. So it will problem start. That's why for quarantine treatment, MBR is more advisable. Suppose if they want, the consumption should be kept for 14 days, madam. Because in the meantime, this egg will hatch into adult larva. So by that time, presence of chemical, it will kill the larva. So 10% mortality will be there, madam. That's
I thank Kolla sir for sharing his experience.
Thank you, sir.
So we can discuss it later. Thank you. So the next speaker is our friend Dr. Prakash sir, and sir is working with ACRP and white grubs since almost more than 10 years, and done a lot of work on leucopolis as well as volotrachea group, and he will be sharing very he did not reveal me on which aspect he is actually talking, and as a result I am also waiting for his talk. So going to Prakash.
Your respected dignitaries and dear friends. So since morning, all the speakers are mercilessly injecting knowledge to already poor students. I think all are exhausted and we need some time to recover. Okay. So I am Prakash, I am working a small scale worker on scarabs. I do So I'll do a little bit of taxonomy and also involved in development of management strategies against major white grubs of Karnataka, and also sharing the knowledge developed in our lab to the stakeholders like NGOs, farmers community, and also amateur biologists. So we are all proud that we belong to the Department of Plant Protection, because the crop division, especially crop protection, we contribute immensely to the production of crops. If our crop protection technologies are implemented at right time, so we can improve the crop productivity to the tune of 30 to 40%. It is proved. And the crop protection technologies are usually developed by the designated scientists. They conduct intensive and extensive studies systematically, and all the technologies they pass through several screening tests, and ultimately they have evolved into a robust package of practices for crop pest. And they develop either crop specific technologies or species specific technologies. This is known factor. But what happens, the majority of times, our crop protection technologies, they fail miserably in the field conditions. That leads to escalation of the crop production cost and also they become root causes for harmful ecological systems. And what could be the reason for this? Ultimately, we blame the poor farmers that he has not adopted our technology in a proper way, or we blame the untimely intervention of the weather parameters and we wash off. Is it true? Is it true? The failure due to only these two factors? Then what? If it is not, then what? So this prompted me to revisit the already developed technologies against white grubs, and also, is there any scope for modification of the technologies, already existing technologies? So what I understood, what I misunderstood, that is, there's a lack of basic knowledge and also poor communication among the scientists itself. So that leads to the miserable failure of our technology in the plant system. So today, Dr. Swami asked me to share some information on the crop protection that related to crop protection of white grubs in a different way. So all of you are aware of that scarab is today morning, many people that talked about scarabs, the pestiferous scarabs, phytophagous scarabs, and so many things. I am not going to talk about the heavy version of scarab, that is taxonomy, biodiversity, all those things, and I hate that all the subjects. So we will talk about the lighter version, or very simple, that is management of white grubs. It is very, very simple. So I take two examples, two beetles. The two beetles have distinct and different biological systems, and how the management differs from each other. So there are two groups. One is Holotricha group, and second one is the Aracanat, commonly known as Aracanat white grub, that belong to genus Leucophilus. There are three species of Leucophilus. All of you know that one. The one is, they are endemic to the Western Ghats and coastal area, and the Holotricha group is predominantly a rain-fed arid soil group. With respect to the elevation, white grub, Aracanat white grubs, they are confined to higher elevations, whereas the Holo group, that is Holotricha group, is confined to the drier part of the country, state. With respect to rainfall distribution, Aracanat white grubs, they predominantly occur in the high rainfall areas of coastal Karnataka and also Malnad region, and all these three species are endemic to Karnataka only. Whereas Holotricha serrata, it is confined to low rainfall areas. That means these two groups, that is Holotricha and also Leucophilus species, they are geographically isolated. So let us see the life of Holotricha serrata. This Holotricha serrata is a polyphagous species. It is a predominant species in South India. And I would like to remind, in the morning we are discussing about the worst range of white grubs. There is no term called worst range for white grubs, because they are polyphagous. So if anybody reports this is new record of white grub, that is nonsense. Okay. So this is the adult beetle. The third instar grub is the most destructive stage of Holotricha serrata, and it causes severe economic losses to many economically important crops like sugarcane, groundnut, even vegetables like beetroot, and commercial crops like onion. And the life of Holotricha serrata begins with the onset of pre-monsoon. It is summer shower, and the life of Holotricha serrata includes 4 stages, as you are all aware. That is a volovateous insect, that is egg, larvae that includes 3 larval stages, 1 pupa, pupal stage, and adult stage. Incubation around 10 to 12 days. The first instar, they completes in 25 to 30 days. Second instar, it takes around 20 to 30 days. Pupa completes the third instar. The third instar is the destructive stage. It is the duration of the third instar is very long, almost 6 to 8 months. It feeds on the live roots of crops. Pupation completes in 15 to 20 days. And there is a conspicuous stage that is called precocious stage, which is reproductively, it is not mature, and it lasts about for 2 to 3 months. Then the reproductively mature adult emerges from the soil during the first shower of the soil, and they land on the nearby tree canopies during the dust cover and go for mating. So this is the biology of Holotrichus serrata. So based on this biology, we have developed management strategies because it feeds on annuals or which is not a short duration plants. So it is better to break the generation in the given location. So it is preventing to become the endemism. So it is the best way is to break the food chain. So what we recommend, a crop rotation with the puddle rice, so that it will break the food supply and also break the generation and helps in the reduction in the population in the given location. So apart from this, based on this biology, we also recommend the larval removal, but the larval removal of larvae from the infested or the affected plant system is practically difficult. And application of insecticide in case of white grub affected field is practically impossible because the symptoms will be noticed after the infestation. By that time, we lose the 90% of the crops. When we see there will be already the 13 star larvae will feeding on the crop, and against the 13 star larvae it is very difficult to go for insecticide application. And right now we don't see any potential biological control agent against white grubs. There is only one option is left over, that is the collection and destruction of adult beetles. Because the Holotrichus serrata, it spends its 90% of the life cycle in the soil. The only visible stage is the adult stage. And the adult stage have some attributes, like the adult, they emerge in mass, then they aggregate on the nearby tree canopies, and the adult emergence is always in synchronous. So these are all the important characteristics. So we can explore these characteristics and advise for the adult beetle collections. But the questions are where to collect adult beetles, when to collect adult beetles, and how long one should go for adult beetle collections. So the operation sex ratio of Holotrichus serrata is unit, that is almost is 1:1. This triggers the synchron in adult emergence on the given day. So after emergence, they aggregate on nearby tree canopies like the neem, the ficus, tamarind, Yunnan mulberry, and folbin, and several different host plants. Here you can see the Holotrichus serrata, it can aggregate on taxonomically unrelated 23 species belonging to 15 families. So this indicates the plant canopy serve as mating platform, and the beetles do not prefer any plant material, any plant species. Okay. The occurrence, that is the emergence across the space, it is not synchronous. So it is vary from the place to place because the adult emergence is triggered by the local edaphic factors. So on the given day, how the adult emergence pattern, it varies. So on the given day, the adult beetle start emerging around 7:30 p.m. Then they land on the tree canopies. Here, the adult beetle emergence was early when the insect population was, the density of the insect population was high. as days, the adult activity pattern, the adult activity window is very long, almost 60 to 80 days. So towards the end of the adult emergence, the arrival is delayed because of the low population density. On the next morning, after emergence, they land on the tree canopy, they go for mating. The mating, it takes place immediately within a few seconds, and the mating duration lasts for 5 to 6 or 7 minutes. Then they detach the male and females. They remain on the tree canopy and remain overnight, then start feeding on the leaves. In the early morning, next morning, they start dispersing by 5:30 a.m. and they go back to the soil for the egg laying. The same process continues for 60 to 80 days, every day, depending upon the soil moisture. So this indicates the adult emerge in the first day and the adult emerge in the last day. They contribute to the generation. Okay, that means all the beetles are important. So what we observed, there is a strong fidelity between the tree canopies, that is harbored beetles during the night hours, and also in the first week itself, there is the number of beetles aggregated tree canopies were very high. With respect to the egg-laying behavior, the adult beetles, they laid most of the eggs, that is more than 50% of the eggs, in the first 10 days itself. So based on the information, what we recommend, adult collection should be done within first week of their emergence. So that gives the more success. So if you delay it by some days, there will not be any profit. So this is lesson we learned. So the lesson what we learned is Olatraca serrata, they aggregated on the 23 species belonging to 13 families. And the adult aggregation behavior indicated on the tree canopies indicated it is only a mating platform, that there is nothing else. And there was a fertility between the tree canopies and also the adult beetles. That means once today some beetles they have aggregated on tree species A or tree number A. Next day also we can expect the same set of beetles aggregated in the same tree only. So that makes us for easy collection destruction of the beetles. I am not going to explain the chemical ecology between these two beetles in this seminar. And also this study provided a lot of information like the mating pattern, fecundity, spatial dispersion, even the presence and absence of the pheromones in the system. So all this information was transferred to our stakeholders for the management of the Olatraca serrata beetle. So this is the one set. So in contrast to Olatraca serrata, we have Leucophalis, which are endemic to the southern part of Karnataka, particularly the coastal and also hilly regions. So Leucophalis conifera, it completes its one generation in 1 year. So Leucophalis vermistri, it takes 2 years to complete its life cycle. This is Leucophalis lepidophora, is the predominant vertical species occurring in the Malnad region. It takes 2 years to complete its life cycle. So what about the management now? Because we are already developed a robust technology, plant protection technology against one species of white grubs. Can we use or can we adopt the same technology to this species also? This is our question. Is it possible? Or should we have a different set of management practices? Then why? So this is the visible symptoms of Leucophalis infested arachnoid garden. Above ground level infestation and below ground infestation, it is self-explanatory. Okay. So here also we have the same set of management practices, but we have some modification. One is collection of adult beetles. And second one is using chemicals or biological agents, the potential biological agents against early historic grubs. And third one is the collection and destruction of grown-up third instar larvae and also yet to emerge adult beetles. But the crucial points are the time, methodology, and the target again. So, in contrast to Holotrichus serrata, here the operational sex ratio is male biased in the field system. So, this triggers adult emergence, but there is no en masse adult emergence. En masse in the sense there is no numerous adult number. In Holotrichus serrata, the number is numerous number, thousands. But in case of Leucophilus species, there are very few adult beetles. And there is no synchrony in the emergence. And there is no definite mating platform. So adult beetles can mate anywhere in the garden, on the ground surface, on the tree canopies, or on the tree trunk. So this makes the collection of adult beetle is difficult in the field. And the adult activity, the adult available in the field is very short. It is almost 8 to 10 days only in case of Leucophilus burmese tree. Whereas in Leucophilus lepidophora, it is staggered emergence. It occupies around 3 to 4 months, but the number of adult available is very, very less. Hardly 8 to 10 days is adult activity period in contrast to Holotrichus serrata. So here, if at all we want to go for adult beetle collection, we should collect within this time period. So the number of adults emerge is low, and the fecundity compared to Holotrichus serrata is less. Almost a female beetle lays 30 or less than 30 eggs compared to Holotrichus serrata. Holotrichus serrata is more than 60 eggs. And also the distribution within the garden is almost aggregate. And it is very difficult to gauge the oviposition site and what is happening in the soil. So what we recommend, the application of insecticide targeting the early instars on the whole garden basis. Okay. So here the expected result is also questionable. And since the areca garden requires a lot of chemicals, the volume of insecticide required is very huge, and ultimately it contaminates our own groundwater and also it reaches the water body and so many environmental issues. So for the past few years, we are deleted this recommendation, that is we are not recommending insecticide application against areca nut oil grubs. And what is the next option? The next option is collection and destruction of grown-up larvae, because the grown-up larvae, they are available in the space and time for the longer time, because the larval duration is almost 16 to 18 months. So digging from the soil and collecting the grown-up larvae, it immediately releases the stress on the plants, and the affected plants immediately put forth its life. So it immediately relieves from the pest attack. So in some time what happens in some gardens, we also come across the overlapping generation insects where along with the grown-up third instar larvae, we see the reproductively matured adults. So if we coincide the operation, we can get the maximum benefit out of it. So we recommend the adult bit, this one, digging and removing of the third instar larva in case of Leucophilus system. So this also indicated a positive impact on the management. You can see here in the first top left, the red indicates the farmers, they have initiated the three-pronged recommendation, the adult beetle collection, the insecticide application, and also collection of larvae. And due to some personal problem, they have given up the operations. And you can see the increase in the population of insect. Whereas the blue one, they continue the operation and there is a reduction in the population. So in case of Conifera, in Conifera, the farmers, they avoided insecticide application. They have strictly followed the adult beetle collection and also removal of larvae. In one farmer, he strictly followed only adult beetle collections, but not on the tree canopy. Before emergence, they identified the time and also they identified the way they emerge from the soil, and they have collected the just emerging the beetles, and they have controlled very successfully. And apart from this, we also noticed the different plant protection, the different plant protection practices adopted by recurrent growers. You can see here the farmers applied the more than the recommended dosage of insecticide. The X indicates the recommended dosage, that is we recommended 1.8, 1.7 liters of chlorpyrifos per acre, but there were 10 times, 10 folds of increase in the dosage. And we recommend insecticide application to only the first or second instar larvae, but majority of the farmers, they targeted the third instar larvae. And we recommend, based on its biology, application on the whole garden basis, but the farmers, they focused only the farm base. And also that led to lot of unconventional methods like application of laterite soil, like lime application and intercropping with banana. But it is for the different reason they grow banana. But some farmers, they assume that banana attracts white grubs and save the arachnoid roots. Ultimately, our operation should be evaluated. So it is almost equal, not satisfied, partially satisfied. So that lead to confusion among the farmers because they have deviated from the recommended the package of practices against the white grubs. Okay. So till I have explained, I have shared the information about the biological system of two distinct white grub species. So let me give some more information about these biological histories. So these Holo and the Leuco groups, they were decided based on the genus, that's Holotrachea, and that's Leucophilus. And Holotrachea, they're distributed in low rainfall areas, whereas Leuco is distributed in high rainfall areas. The adult emergence is synchronous. Adult emergence synchronous on the given day. Between the sexes also it is synchronous. Whereas in case of Leucophilus, it is asynchronous. That means adult beetles, they did not emerge in synchrony on the given day. Usually, the Holotrachea serrata beetle, they emerge around 7:30 p.m., whereas Leucophilus usually emerge around 6:40 p.m. But during overcast condition, the Leucophilus species, they emerge in the daytime also. So that leads to a lot of confusion among the farmers. And the mating, Holotrachea serrata is polyandrous. There is a multiple mating. Whereas in case of Leucophilus, female mates only once. And adult sex ratio is 1:1, whereas it is adult sex ratio. The adult sex ratio in case of Leucophilus, female biased. But on a given day, it becomes a male biased. And the growth consumption rate in the Holotrachea serrata is relatively faster because they feed on the short duration resources, so they feed very fast. Whereas Leucophilus, the feeding rate is relatively slow because the resource is surplus and available around the year. The population, the spatial distribution of the population in case of this one, Holotrachea serrata, it is wider. It is always spreading its territory. Whereas leucophilis, it is very narrow. The diet breadth of Voloteca serrata is wider, and the diet breadth of leucophilis is very narrow. So early morning we are discussing about the diet breadth of white grubs. Okay. And usually Voloteca serrata that feeds on annuals, or the grubs feed only for 4 to 5 months. Whereas in leucophilis, the grub, it feeds almost 8 to 10 months. It's a long duration feeding stage. And adults mating, as I mentioned, in case of Voloteca serrata, there is a definite mating platform. In case of leucophilis, there is no definite mating platform. So, in case of Voloteca serrata, the females, they may not produce greater pheromones. But in case of leucophilis, the females tend to produce greater pheromones. And we can see, since in case of Voloteca serrata, it is 1:1 sex ratio, we don't find the male chasing behavior. In case of leucophilis, we find the male chasing behavior because of the sex ratio, biased sex ratio. And within night adult emergence, there is synchrony without any difference between the sexes in case of Voloteca serrata. That means male and female, they emerge together. But in case of leucophilis, usually it is a fraternity, that is males, they emerge first. That's why we tend to collect male beetles only, and we claim that it is male biased. But it is not male biased in general. Ultimately what happens, the variation they accumulated during its developmental period will nullify at one stage, one point of time. So that we termed as forward break. That creates a discrete generation in case of Voloteca serrata, whereas the protandry, it drives the discrete generation in case of leucophilis, leucophilis species. So this is the life of these two insects. Based on this information, we formulated the plant protection measures. So this suggests for any plant protection scientist, before formulating any plant protection measures, one should understand the insect completely, holistically, and go for the development of the measures. Otherwise, they fail miserably in the plant system, and we blame the stakeholders. So, we should always address the resource consumer relationship and also the mating pattern of our studied the insects for development of the robust plant protection technology, because the life histories dictate the pest management strategies. Thank you. Now, if you have any clarification, I am ready to answer. Mike, what would you say?
Compared between the Voloteca as well as the leucophilis one species, what happens to the other two species? Is that the behavior similar with respect to the fraternity or maybe the male-female ratios and then migration? Whatever is it?
Leucophilis, lepidophoran, burmistry, Leucophilis, lepidophoran, burmistry, they are more or less same. But Coniophora, it is annual. But the biological parameters are almost the same. But Leucophilis coniophora, it resembles in somewhere Voloteca serrata. So that is why it is difficult. See, I have taken 8 years to understand this one. Still, I have to understand many factors.
Tell us where exactly the Coniophora is.
Coniophora, it is coastal. See, distribution pattern is like this. Leucophilis lepidophora is the predominant species from hilly area, right from Sirsi to your Tirthalli zone. And in some parts of Maharashtra, it also feeds on sugarcane. Whereas Coniophora is limited to coastal, and it is a predominant white grass species in Kerala. It feeds on areca nut, coconut, and also Topiaca. But the distribution of Burmese tree is highly variable. It is scattered between low and also high elevation. It is very, very difficult. And morphologically, Lepidophora is distinct. But Coriophora and Burmese tree are morphologically indistinguishable. This is the point. What you other species means? There are 2,000 species of white grubs in India, and they are geographically isolated. So I have taken 10 years to understand only one insect species. If I start on morning, I am thankful to Dr. Swami. While addressing, he reduced my age by 20 years. He said all are almost 30 years. Thank you, Kalesh. So in North India, we have 3 species. One is Anomala dimidiata, is sub-Himalayan species. Second is Holotricha consanguinea, it is also North Indian species. And we have few Lepidiota species, Lepidiota mansueta, Lepidiota alti stigma. They are confined to northeastern zone. In the central zone, we have different groups of Holotricha, and South India is predominated by Holotricha serrata. And people have also reported Holotricha rhinodi, Holotricha fissa, Soprops, Miridiba, so many things. But for your information, for the information of the body, I would like to tell you, identification of scarabs is very, very difficult. And people are reporting, I don't know how exactly they are reporting, because there is very difficult to access the types. Very difficult to access the types. Still we don't know what is Holotricha rhinodi. Holotricha rhinodi is native of Sri Lanka. It is a native of Sri Lanka, described from Sri Lanka. Still date we don't know what is Holotricha rhinodi. And recently, people are reporting. So don't blame scientists. There's a lot of problem. So see, ambiguity is due because the first thing is See, another thing is I have started working on Holotricha group. I have started during my MSc period some 30 years back. Now I have once again initiated the taxonomy studies on Holotricha to delimit the species Holotricha serrata in India. So scientists working on scarabs, they are reporting Holotricha species, Holotricha serrata from all over India. But we don't know what is Holotricha serrata. And now I am working with 5 international collaborators. Recently what I heard, they are unable to trace the type. We don't know where is the type is there. In 1850, Blanchard reported Holotricha, this group from Mysore. Another insect I will tell you. One species, not Holotricha, Shivappa. He has reported in 1850. This is for the sake of my students. So what happens, we don't address the issues rationally. Early morning, Dr. Kalesh also is mentioning what happens if we don't give attention towards the literature and also distribution pattern of species. In 1850, Blanchard reported one species from Mysore. It is in German. In the next issue, another person has reported the same species from the North India. In the next issue, another person, he has published the same species from Mysore, Karnataka. So I have been working on white grubs for the past 20 years, And in our survey, I could not encounter a single beetle of that species from Karnataka. But fortunately, we have collected, Dr. Yashwant has collected the same species from Kodaikanal. So now tell me, what is the type locality? What is the type locality? We don't know type locality. For your Lycophilis also, types are not available. So I have been struggling to get a Lycophilis type since 1 decade. We don't know, the Burmester he has deposited somewhere else. We don't know. So this is happens. So we have to go.
Okay, finally, we have
No, but they have reported from Belagaum, but I have not collected from Belagaum.
we have.
It is North Indian species.
So,
See, one thing one should remember, see the presence and absence of species that depends on our intensive collections. And if I say that it is not available in the one place, that doesn't mean that it's not available. That indicates that I have not thoroughly examined, that's all. And another thing is, there is a latitudinal effect on the distribution pattern of organisms. Okay, so that also restricts the spatial distribution of organisms. But some species like leafhopper, they are cosmopolitan. So that happens. So the theme of my seminar is, so generalization is not possible. Each species is distinct. Each species is distinct, unique and indispensable. So respect the species. Okay. Only 3 questions. Tell me, sir.
So I know you mentioned that you don't want to touch chemical technology, but I need to pick on that. And you mentioned that you have also identified given to the stakeholders for the whole country.
We tried our level best, even though we know that there is the pheromone, presence of pheromone is remote. Okay, we tried. One of my senior he tried his level best, even though we know that it is remote. But Kaleshwar Swami also tried, and he developed from Lycophilis lepidophora, but some problem is there. But recently I have isolated pheromone from Lycophilis burmisteri, that it is going on. Just to understand the pheromone is present or absent, I have taken 6 years. Holotrichia, it is strong aggregation pheromones, but it is high molecules, we don't know. We tried our level best. Consanguinea, it is different set of story. It is total different set of story. It is female released semi-chemicals. They did not termed sex pheromones in 1965.
Since you are like looking at the pheromone in Lycophilis, did we give a trial on its own and tested it on its own?
We tried, we tested. It is purely sex pheromone in case of Lycophilis. 100%. Okay, sir. Raghu. Sir, it is difficult to say because white grubs, that doesn't give any room for any kind of management. They produce very stable population. Very stable population. So that is the biggest problem in case of management. So even they can tolerate significantly, they can avoid this, successfully avoid from this biological system. But I still hope for biological control. Even if any biological control agent which can give 30% mortality, I am ready to accept it. 30% is sufficient. But in the confined system, it's okay. But in the field situation, it is.
Practically,
Yes. That.
Looking for alternative management strategies like
So we have to follow the insect. Insect will not follow us. I take this opportunity to thank Dr. Kaleshwar for remembering me and give me an opportunity to share my knowledge, share my few information with you. Thank you very much. And I also thank the University of Agriculture and Horticulture Sciences and all my seniors. Thank you very much. Thank you, sir.
Last thing we have Ishanya Sharma. Okay, so the most youngest among the speakers. Speaking on insect photography, but in meantime we told that we mix both English and Kannada. So basically from Shivamogga, Sagar. I got no problem at all because photo will speak. So he's a BCom graduate and no wildlife photographer. Okay, so that's how it will be most interesting for students, and then now as they use mobile itself, so probably the ways, the important gadgets which can be used for photography.
I have also included spiders here because the method of spiders and insects photographs are same. So, and I also don't take much time because already time is over. So, let's start. Let's start with the required camera equipments. Macro lenses, flashes, diffusing to the filter. I will tell one by one. Yeah, okay. Macro lens. Insect photography might need macro lens for macro lens ತುಂಬಾ ಕ್ಲೋಸ್ ಅಲ್ಲಿ ತೋರ್ಸಕ್ಕೆ ಬೇಕಾಗುತ್ತೆ. Robber fly feeding on bottle fly. This is a horse fly. Macro lens ಇರೋದ್ರಿಂದ ನಿಮಗೆ ಈಸಿಯಾಗಿ ಅಪ್ರೋಚ್ ಮಾಡ್ಬೋದು ಫೋಟೋ ತೆಗೆಯೋಕ್ಕೆ. ಬೇರೆ ಮೊಬೈಲಲ್ಲೆಲ್ಲ ಆದರೆ ನಾವು ಅದನ್ನು ಫೋಟೋ ತೆಗೆಯೋ ಅಷ್ಟೊತ್ತಿಗೆ ಅದು ಹೋಗಿರುತ್ತೆ. ದಿಸ್ ಈಸ್ EPS ಜಂಪಿಂಗ್ ಸ್ಪೈಡರ್. ಇದನ್ನು ನಾನು ಕ್ಯಾಪ್ಚರ್ ಮಾಡಿದ್ದು ನಿಕಾನ್ D7200 ವಿತ್ ಮ್ಯಾಕ್ರೋ ಲೆನ್ಸಿಂದ. ಇದು ಮ್ಯಾಂಟಿಡ್ ಫ್ಲೈ. ದಿಸ್ ಈಸ್ ಥ್ರೋನ್ ಮಿಮಿಕ್ ಪ್ಲಾಂಟೋಪರ್ ನೇಮ್. This is leaf insect. Another equipment we need for macro photography is flash. Flash ಇರೋದ್ರಿಂದ ನಮಗೆ ನೈಟ್ ಫೋಟೋಗ್ರಫಿ ಮಾಡ್ಬೋದು. ಆ್ಯಂಡ್ ಆಲ್ಸೋ ನೀವು ನಾವು ಸಣ್ಣ ಇನ್ಸೆಕ್ಟ್ ಫೋಟೋ ತೆಗಿಬೇಕು ಅಂತ ಅಂದಾಗ ಏನಾಗುತ್ತೆ ಅಂದರೆ ನಾವು ನ್ಯಾಚುರಲ್ ಲೈಟ್ ಕಟ್ ಆಗುತ್ತೆ ಫೋಟೋ ತೆಗಿತಾ ಇರಬೇಕಾದ್ರೆ. ಸೊ ಫ್ಲ್ಯಾಶನ್ನು ಯೂಸ್ ಮಾಡೋದ್ರಿಂದ ನಮ್ಗೆ ಡೀಟೇಲ್ ಆಗಿ ತೆಗಿಬೋದು ಫೋಟೋನ. ಇದು ಕೋಕೋಲಸ್ ಜಂಪಿಂಗ್ ಸ್ಪೈಡರ್ ಅಂತ. ಇದೊಂದು 5 to 6 mm ಇರುತ್ತೆ. ಸೊ ಫ್ಲ್ಯಾಶ್ ಯೂಸ್ ಮಾಡಿರೋದ್ರಿಂದ ನಿಮಗೆ ಡೀಟೇಲ್ ಆಗಿ ಕಾಣ್ತಾ ಇದೆ ಈ ಫೋಟೋ. ದಿಸ್ ಈಸ್ ಆ್ಯಂಡ್ ಆಲ್ಸೋ ಅದೇ ಈ ಥರ ಸಣ್ಣ 3-4 mm ಇನ್ಸೆಕ್ಟ್ಸನ್ನು ತೆಗಿಬೇಕಿದ್ರೆ ನ್ಯಾಚುರಲ್ ಲೈಟ್ ಕಟ್ ಆಗೋದ್ರಿಂದ ಅದೇ ಫ್ಲ್ಯಾಶ್ ಹಾಕಿದಾಗ ನಿಮಗೆ ಅದು ಶಾರ್ಪ್ನೆಸ್ ಮತ್ತು ಡೀಟೇಲ್ ಆಗಿ ಸಿಗುತ್ತೆ. ಆ್ಯಂಡ್ ಫೈರ್ ಫ್ಲೈ ಲಾರ್ವಾ ಫೀಮೇಲ್. ಇದು ಹಾಂ ಓಕೆ. ದಿಸ್ ಈಸ್ ಮೇ ಬಿ ಸಮ್ ಲೈಸ್ nymph. ಹಾಂ wingless this is maybe emerald moth diffuser ನೆಕ್ಸ್ಟ್ ಈಗ ಫ್ಲ್ಯಾಶು ಮತ್ತು ಮ್ಯಾಕ್ರೋ ಲೆನ್ಸ್ ಆದಮೇಲೆ ಮೇನ್ ಇಂಪಾರ್ಟೆಂಟ್ ಬೇಕಾಗಿರೋದು ಡಿಫ್ಯೂಸರ್. ಯಾಕಂದರೆ ನೀವು ಡೈರೆಕ್ಟಾಗಿ ಫ್ಲ್ಯಾಶ್ ಹಾಕಿದರೆ ಒಂದು ಹಾರ್ಶ್ ಆಗುತ್ತೆ ಫೋಟೋ, ಮತ್ತು ಇನ್ನೊಂದು ತುಂಬ ಡಿಸ್ಟರ್ಬ್ ಆಗುತ್ತೆ. ಈಗ ಡಿಫ್ಯೂಸರ್ ಆಗೋದ್ರಿಂದ ಸ್ವಲ್ಪ ಹಾರ್ಮನ್ನು ಕಮ್ಮಿ ಹಾರ್ಮ್ ಆಗುತ್ತೆ. ನೋಡ್ಬೋದು ವಿತೌಟ್ ಡಿಫ್ಯೂಸರ್, ವಿತ್ ಡಿಫ್ಯೂಸರ್. ಫಸ್ಟ್ ಫೋಟೋ ಡೈರೆಕ್ಟ್ ಫ್ಲ್ಯಾಶ್ ಹಾಕಿದ್ದು, ಲೈಟ್ ಕಣ್ಮೇಲೆ ಬಿದ್ದಿದೆ. ಸೊ ಡಿಫ್ಯೂಸ್ ಮೇಕ್ ಫೋಟೋ ಬ್ಯೂಟಿಫುಲ್. ಡೋಂಟ್ ನೋ ವಿಚ್ ಮೋತ್ ಈಸ್ ಇಟ್. ಡಿಫ್ಯೂಸರ್ ಯೂಸ್ ಮಾಡಿರೋದ್ರಿಂದ ಲೈಟೆಲ್ಲ ಅವಾಯ್ಡ್ ಆಗಿ ನೀಟಾಗಿ ಬಂದಿದೆ. ಸಮ್ ಶೀಲ್ಡ್ ಬಗ್ ಮೇಲೆ again and another one is close-up filter. ನಾವು ಅಲ್ಟ್ರಾ ಕ್ಲೋಸಪ್ ಫೋಟೋಸ್ ತೆಗಿಬೇಕು ಅಂದರೆ ನಮಗೆ ಕ್ಲೋಸಪ್ ಫಿಲ್ಟರ್ ಬೇಕು. ಡೈರೆಕ್ಟ್ ಕ್ರಾಪ್ ಫೋಟೋ ಮ್ಯಾಕ್ರೋ ಲೆನ್ಸಲ್ಲಿ ಕ್ರಾಪ್ ಮಾಡಿ ತೆಗೋಕ್ಕಿಂತ ಕ್ಲೋಸಪ್ ಫಿಲ್ಟರ್ ಯೂಸ್ ಮಾಡಿದ್ರೆ ನಿಮ್ಗೆ ಇನ್ನೂ ಡೀಟೇಲಾಗಿ ಬರುತ್ತೆ. ಇದು ಸ್ನೇಲ್ ಎಕ್ಸ್, ಇದಕ್ಕೆ ಡಿಫ್ಯೂಸರ್ ಮತ್ತು ಫ್ಲ್ಯಾಶ್ ಮ್ಯಾಕ್ರೋ ಲೆನ್ಸ್ ಎಲ್ಲ ಯೂಸ್ ಮಾಡಿ ತೆಗೆದಿದ್ದು. ಇದು ಮೇ ಬಿ ಒಂದು 5 mm ಇರುತ್ತೆ. ಸೊ ಕ್ಲೋಸಪ್ ಫಿಲ್ಟರ್ ಯೂಸ್ ಮಾಡಿರೋದ್ರಿಂದ ನಿಮಗೆ ತುಂಬ ಕ್ಲೋಸ್ ಆಗಿ ಕಾಣಿಸುತ್ತೆ. ದಿಸ್ ಈಸ್ ಇಂಡೋಪೆಡೆಲ್ಲ ಜಂಪಿಂಗ್ ಸ್ಪೈಡರ್ ಫೀಮೇಲ್. ದಿಸ್ ಈಸ್ ಜ್ಯುವೆಲ್ ಬೀಟಲ್. ಸಮ್ ಲೀಫ್ ಬಗ್ ನಿಂಪ್ ಮೇ ಬಿ ಇದು. ಮತ್ತು ಈ ಥರ ಸಣ್ಣ ಹುಳಗಳನ್ನು ತೆಗೆಯೋಕ್ಕೂ ಅದು ಹೆಲ್ಪ್ ಆಗುತ್ತೆ. ಕ್ಲೋಸಪ್ ಫಿಲ್ಟರ್ ಯೂಸ್ ಮಾಡೋದ್ರಿಂದ ತುಂಬ ಸಣ್ಣ ಕೀಟಗಳನ್ನು ನಾವು ಕ್ಲೋಸಾಗಿ ತೋರಿಸ್ಬೋದು. ಬಾರ್ಕ್ಲೈಸ್ ಮೇ ಬಿ. ದಿಸ್ ಈಸ್ ಟೈಗರ್ ಬೀಟಲ್. ಇದಕ್ಕೆ ನಾನು ಕ್ಲೋಸಪ್ ಫಿಲ್ಟರ್ ಯಾಕೆ ಯೂಸ್ ಮಾಡಿದ್ದು ಅಂದರೆ ಆ ವಾಟರ್ ಡೀಟೇಲ್ ಮತ್ತು ಐ ಡೀಟೇಲ್ನ ತೋರಿಸುತ್ತೆ. ಪೇಪರ್ ವ್ಯಾಸ್, ಸಮ್ ಕ್ರಿಕೆಟ್. ಯು ಕ್ಯಾನ್ ಆಲ್ಸೋ ನೀವು ಇಲ್ಲಿ ಇದನ್ನು ನೋಡ್ಬೋದು, ಇದುವೇ ಇದೆ, ಎಂಟಿದೆ ಕ್ರಿಕೆಟ್ ಮೇಲೆ. ನೆಕ್ಸ್ಟ್ ಈಗ ಬಡ್ಜೆಟ್ ಫ್ರೆಂಡ್ಲಿ ಕ್ಯಾಮ್ರಾ, ಅಂದರೆ ಈಗ ಬಡ್ಜೆಟ್ ಇಲ್ಲ ತಗೊಳ್ಳೋಕ್ಕೆ ಅಥವಾ ಜಸ್ಟ್ ಫಾರ್ ರೆಕಾರ್ಡಿಂಗ್ ಪರ್ಪಸ್ ನೀವು ಕ್ಯಾಮ್ರಾ ಯೂಸ್ ಮಾಡ್ತೀರಾ ಅಂದರೆ ಪಾಯಿಂಟ್ ಆ್ಯಂಡ್ ಶೂಟ್ ಕ್ಯಾಮ್ರಾ ಇದೊಂದು 5,000ದಿಂದ ಸಿಗುತ್ತೆ ನಿಮಗೆ. ಇದು ನಿಕಾನ್ ಕೂಲ್ಫಿಕ್ಸ್ 5300 ಅದರಿಂದ ತೆಗೆದಿದ್ದು. ದಿಸ್ ಒನ್ ಆಲ್ಸೋ ಇಸ್ ಕ್ಯಾಪ್ಚರ್ಡ್ ವಿತ್ ಕೆನಾನ್ ಪವರ್ ಶಾಟ್. ಇದು ರೆಡ್ ಸ್ಪಾಟೆಡ್ ಡ್ಯೂಕ್ ಕ್ಯಾಪ್ಚರ್ಡ್ ವಿತ್ ನಿಕಾನ್ ಕೂಲ್ಫಿಕ್ಸ್ P900. ಲೈಕನ್ ಸೆನ್ಸ್ಮ್ಯಾನ್ ಸ್ಪೈಡರ್. ನೆಕ್ಸ್ಟು ನೀವು ಬೇಕಿದ್ರೆ ಮೊಬೈಲಲ್ಲೂ ಮ್ಯಾಕ್ರೋ ಲೆನ್ಸ್ ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಫೋಟೋನ ಬ್ಯೂಟಿಫುಲ್ ಆಗಿ ತೆಗಿಬೋದು. ಬಟ್ ನಾನು ಯೂಸ್ ಮಾಡಿಲ್ಲ, ಸೊ ನನ್ನ ಫ್ರೆಂಡ್ ಯೂಸ್ ಮಾಡಿರೋ ಒಂದಿಷ್ಟು ಫೋಟೋಸ್ ತೋರಿಸ್ತೀನಿ. ಸುನೀಲ್ ಕುಮಾರ್ ಅಂತ, ಈಸ್ ಗ್ರೇಟ್ ಮ್ಯಾಕ್ರೋ ಮೊಬೈಲ್ ಫೋಟೋಗ್ರಾಫರ್. ನೆಕ್ಸ್ಟ್ ಲರ್ನ್ ಆ್ಯಂಡ್ ಕ್ಯಾಪ್ಚರ್ ಬಿಹೇವಿಯರ್ನ ಲರ್ನ್ ಮಾಡೋದ್ರಿಂದ ನಾವು ಫೋಟೋಗ್ರಫಿನ ಇನ್ನೂ ಇಂಟ್ರೆಸ್ಟಿಂಗ್ ಆಗಿ ತೋರಿಸ್ಬೋದು. This is maybe pumpkin beetle mating, fruit fly mating. ಇದು ಸ್ಟಿಂಕ್ ಬಗ್ಗೆ ಜಸ್ಟ್ ಹ್ಯಾಚ್ ಆಗಿರೋದು ನಿಮ್ಸು ವಿತ್ ನೀವು ಎಕ್ಸು ನೋಡ್ಬೋದು ಮೊಟ್ಟೆಗಳು. ಇದು ಪೈರೇಟ್ ಸ್ಪೈಡರ್ ಅಂತ. ಈ ಸ್ಪೈಡರ್ ಬರೀ ಸ್ಪೈಡರನ್ನು ಕ್ಯಾನಿಬಲ್ ಸ್ಪೈಡರ್, ಇದು ಬರೀ ಸ್ಪೈಡರನ್ನು ಮಾತ್ರ ತಿನ್ನುತ್ತೆ. ಮತ್ತು ಇದು ಸ್ವಂತವಾಗಿ ಇದಕ್ಕೆ ಬಲೆ ಅಷ್ಟು ಬಿಡ ಎಬಿಲಿಟಿ ಇರಲ್ಲ. ಸೊ ಇದು ಬೇರೆ ವೆಬ್ಬನ್ನು ಪೈರೇಟ್ ಮಾಡುತ್ತೆ. ಸೊ ಇದನ್ನು ಪೈರೇಟ್ ಸ್ಪೈಡರ್ ಅಂತ ಕರೀತಾರೆ. ಇದು ಮತ್ತೆ ಸ್ನಿಂಕ್ ಬಗ್ ವಿಚೆಕ್. ಇದು ಜಂಪಿಂಗ್ ಗ್ಯಾಂಟ್. ಇದು ಅಷ್ಟೇ, ಇದು ತುಂಬ ಸ್ಪೀಡ್ ಇರುತ್ತೆ. ಸಿಕಾಡ ಮಾಲ್ಟಿಂಗ್ ಆಗ್ತಾ ಇರೋದು. Meripanthus spider feeding on link spider. ಇದು ಇಲ್ಲಿ entomopathogenic fungus infected green bottle fly maybe. Another one is ant mimic spider. Weevil bug mating. ಪ್ಲೇ ವಿತ್ ಲೈಟ್ಸ್. ಈಗ ನಾವು ಬೆಳಕಿನ ಜೊತೆ ಅದೇ ಬೇರೆ ಡಿಫ್ರೆಂಟ್ ಆ್ಯಂಗಲಿಂದ ಲೈಟ್ ಕೊಡೋದ್ರಿಂದ ಪಿಕ್ಚರನ್ನು ಇನ್ನೂ ಅಟ್ರಾಕ್ಟಿವ್ ಆಗಿ ನಾವು ತೋರಿಸ್ಬೋದು. ಮೇ ಬಿ ವೈನ್ ಮಾತ್. ಇದು ಸನ್ಲೈಟ್, ಬ್ಯಾಕ್ ಸನ್ಲೈಟಿಂದ ಫೋಟೋ ತೆಗೆದಿದ್ದು. ಸಮ್ ಕ್ಯಾಟಿಡ್ ಎಕ್ಸ್ಟರ್ನಲ್ ಔಟ ಕ್ಯಾಮ್ರಾನ ಫ್ಲ್ಯಾಶನ್ನು ಎಕ್ಸ್ಟರ್ನಲ್ ಕೊಟ್ಟು ತೆಗೆದಿದ್ದು ಮೇಲೆ ಟಾಪ್ ಆ್ಯಂಗಲಿಂದ ಬ್ಯಾಕ್ ಲೈಟ್. ಕ್ಯಾಟಿಡ್ ಮಾಲ್ಟಿಂಗ್ ಬ್ಯಾಕ್ ಲೈಟ್. assassin bug feeding on some wolf spider. This again catidid feeding, praying mantis feeding on some fly, don't know which one is. Flower mantis, and I used backlight for that beautiful flame. ಇದು ಬ್ರ್ಯಾಂಕಸ್ ಕಲಾಬಿ ಅಂತ ಜಂಪಿಂಗ್ ಸ್ಪೈಡರು. ಇದಕ್ಕೆ ನಾನು 2 ಫ್ಲ್ಯಾಶ್ ಯೂಸ್ ಮಾಡಿದ್ದೀನಿ. ಒಂದು ಆನ್ ಕ್ಯಾಮರಾ ಫ್ಲ್ಯಾಶು, ಇನ್ನೊಂದು ಕೆಳಗಡೆಯಿಂದ ಇನ್ನೊಂದು ಫ್ಲ್ಯಾಶ್ ಕೊಟ್ಟಿದ್ದೀನಿ. ನೀವು 2-3 ಜನ ಇದ್ದಾಗ ಈ ಥರದ್ದು ಕ್ರಿಯೇಟಿವ್ ಟ್ರೈ ಮಾಡ್ಬೋದು. ಇದು ಅಟ್ಲಾಸ್ ಮೌತ್ ಕ್ಯಾರೆಕ್ಟರ್ ಪಿಲ್ಲರು. ಕ್ಯಾಪ್ಚರ್ ದೆಮ್ ಇನ್ ಆ್ಯಕ್ಷನ್, ಟ್ರೈ ಟು ಕ್ಯಾಪ್ಚರ್ ದೆಮ್ ಇನ್ ಆ್ಯಕ್ಷನ್. ಇದು ಟೈಲ್ಗೆ ಫ್ಲೈಯಿಂಗ್ ಮಾಡ್ತಾರೆ. ಇದು ಸೌದರ್ನ್ ಬರ್ಡಿಂಗ್ ಫಿಮೇಲನ್ನು ಅಟ್ಯಾಕ್ ಮಾಡ್ತಾರೆ. ಇದು ಸೈಲರ್ ಜಂಪಿಂಗ್ ಸ್ಪೈಡರ್ ಅಂತ. ಇದು ಆ್ಯಂಟ್ ಮಿಮಿಕ್ ಸ್ಪೈಡರು. ಜಸ್ಟ್ ಈಗ ಬಲೆ ಬಿಟ್ಟು ಕ್ಯಾಚ್ ಮಾಡಿದೆ, ಆ ಟೈಮಲ್ಲಿ ತೆಗೆದಿತ್ತು. ಟ್ರೈ ಎ ಡಿಫ್ರೆಂಟ್ ಫ್ರೇಮ್. ನೀವು ಒಂದು ಸಬ್ಜೆಕ್ಟನ್ನು ನೋಡಿದಾಗ ಫ್ರೇಮನ್ನು ನೀವು ತಲೆಯಲ್ಲೇ ಈ ಥರ ತೆಗಿಬೇಕು ಅಂತ ಒಂದು ಥಿಂಕ್ ಮಾಡಿ. Praying mantis with sunset bouquet. Maybe monkey puzzle butterfly. I placed it 1/3 frame. So maybe throne mimic plant hopper nympho. Snail, hummingbird moth caterpillar. Weevil bug. ಇದು ರನ್ನಿಂಗ್ ಕ್ರಾಫ್ಟ್ ಸ್ಪೈಡರ್ ಅಂತ ಟರ್ಮೈಟ್ ಫೀಡ್ ಮಾಡ್ತಾರೆ. ಮತ್ತೆ ಅದೇ ರನ್ನಿಂಗ್ ಕ್ರಾಫ್ಟ್ ಸ್ಪೈಡರ್. ಅನದರ್ ಸಮ್ ಪ್ಲಾಂಟ್ ಹಾಪರ್ ಮೇ ಬಿ. I used external flash for this, and shield bug. ಆಲ್ಸೋ ಡಿಫ್ರೆಂಟ್ ಆ್ಯಂಗಲಿಂದ ನೀವು ಫೋಟೋಗ್ರಫಿ ಮಾಡೋದ್ರಿಂದ ಐಡೆಂಟಿಫಿಕೇಶನ್ ನೀವು ಹೆಲ್ಪ್ ಆಗುತ್ತೆ. ಲಾಂಗ್ ಆ್ಯಂಡ್ ಬೀಟರ್. ಇದು ರಾಬರ್ ಫ್ಲೈ, ಗ್ರೀನ್ ಬಟರ್ಫ್ಲೈನ ಫೀಡ್ ಮಾಡ್ತಾ ಇರೋದು. ಇದು ವೈಲಿನ್ ಮ್ಯಾಂಟಿಸ್ ಅಂತ. ಕ್ಲೋಸರ್ ಆ್ಯಂಡ್ ವೈಡ್ ಆ್ಯಂಗಲ್. ಮತ್ತು ಬಿ ಪೇಷನ್ಸ್ ಅಂದರೆ ನೀವು ಒಂದು ಸಬ್ಜೆಕ್ಟಿಗೆ ಕಾಯಬೇಕು ಸಿಗೋ ತನಕ. ಇದು ಕ್ರಿಸಿಲ್ಲಾ ಜಂಪಿಂಗ್ ಸ್ಪೈಡರ್ ಅಂತ, ಇದೊಂದು 4 to 5 mm ಇಂಚ್ ಇರುತ್ತೆ, ತುಂಬ ಸಣ್ಣ ಇರುತ್ತೆ. ಇದು ಗ್ರೌಂಡ್ ಲೆವೆಲಲ್ಲಿ ಸಿಗೋಂಥ ಸ್ಪೈಡರು. ಇದು ತುಂಬ ಸ್ಪೀಡಾಗಿ ಓಡುತ್ತೆ. ಇದೊಂದು ಫೋಟೋ ತೆಗೆಯೋಕ್ಕೆ ನಾನು ಒಂದು 20 to 30 ಅಟೆಂಪ್ಟ್ಸ್ ನನಗೆ ಒಳ್ಳೆ ಫೋಟೋ ಸಿಕ್ಕಿರಲಿಲ್ಲ. ಇದು ಓಕೆ ರೆಕಾರ್ಡ್ ಫೋಟೋ ಸಿಕ್ಕಿದೆ. ಆ್ಯಂಡ್ ಆಲ್ಸೋ ಮೇ ಬಿ ಸ್ಪೈಡರ್ ಮ್ಯಾನ್ ಇನ್ಸ್ಪೈರ್ಡ್ ಬೈ ದಿಸ್ ಸ್ಪೈಡರ್. ಇದು ಸ್ಪೈಡರ್ ವ್ಯಾಸ್. ಇದು ಅಷ್ಟೇ ಸಿನೋಮೆಸಿಸ್ ಕ್ರಾಪ್ ಸ್ಪೈಡರ್ ಅಂತ. ಇದು ಒಂದು ಫ್ಲೇವರ್ ಒಳಗಡೆ ಕೂತ್ಕೊಂಡಿರುತ್ತೆ. ಬಿ ಬರೋಂಥ ಫ್ಲೇವರ್ ಒಳಗಡೆ ಅದು ಬಿ ಬರೋ ತನಕ ಕಾಯುತ್ತೆ, ಕಾದು ತಿನ್ನುತ್ತೆ. ಸೊ ಅದು ಕಾಯೋದ್ರೊಳಗೆ ನಾವು ಕಾಯಬೇಕು ಅದ್ರ ಜೊತೆಗೆ. ಈ ಥರದ್ದೊಂದು ಫೋಟೋಸ್ ಬೇಕಂದರೆ ಇದು ಲಾಸ್ಟ್ ಪೋರ್ಷಿಯಾ ಸ್ಪೈಡರ್ ಅಂತ. ಇದು ಮೋಸ್ಟ್ ಇಂಟೆಲಿಜೆಂಟ್ ಸ್ಪೈಡರು. ಇದು ಏನು ಮಾಡುತ್ತೆ ಅಂದರೆ ಇದು ಸ್ಪೈಡರನ್ನು ಮಾತ್ರ ತಿನ್ನೋದು. ಸ್ಪೈಡರ್ ಇದ್ರ ಪ್ರೇ ಇರುತ್ತಲ್ಲ, ಅದು ಯಾವ ಇದು ಇನ್ಸೆಕ್ಟ್ ಅಂತ ತಿನ್ನಲ್ಲ, ಆದರೂ ಇದು ಇನ್ಸೆಕ್ಟ್ ಹತ್ರ ನೆಸ್ಟ್ ಮಾಡುತ್ತೆ, ಅಂದರೆ ವೆಬ್ ಹಾಕಿ ಅಲ್ಲಿ ಸ್ಟೇ ಮಾಡುತ್ತೆ. ಯಾಕಂದರೆ ಆ ಇನ್ಸೆಕ್ಟನ್ನು ತಿನ್ನಕ್ಕೆ ಬರೋ ಪ್ರೀಡಿಯೇಟರು ಇದ್ರ ಪ್ರೇ ಆಗಿರುತ್ತೆ. ಸೊ ಅದು ಬಂದು ಇದು ವೆಬ್ ಬಿಲ್ಡ್ ಮಾಡೋ ತನಕನೂ ಕಾದು ಬಿಲ್ಡ್ ಮಾಡಾದ್ಮೇಲೆ ನಿಧಾನ ಅದ್ರ ವೆಬ್ಬಿಗೆ ಇದು ಹೋಗಿ ಪ್ರೇ ಥರ ಹೋಗುತ್ತೆ. ಅದು ಇದನ್ನು ಹಿಡಿಯಕ್ಕೆ ಬಂದಾಗ ಅದನ್ನು ಹಿಡಿಯುತ್ತೆ. ಸೊ ಇದು ಎಲ್ಲ ಫೋಟೋ ನಾನು ತೆಗೆದಿದ್ದು ಬ್ಯಾಕ್ಯಾರ್ಡ್ ನಮ್ಮ ಮನೆ ಸುತ್ತಮುತ್ತ. ಥ್ಯಾಂಕ್ ಯು. Thank you, Dr. Kaleshwar Swami sir, for opportunities, and Shara sir.
So we fulfill the professor's question, that will be restrict question only for students. One question per
Ishan, how come you know every name of all the insects?
I have interest, so ಇಲ್ಲ ಕೇಳ್ತೀನಿ ಗೊತ್ತಿದ್ದವ್ರ ಹತ್ರ. ಐ ವಿಲ್ ಆಸ್ಕ್. ಮತ್ತೆ ಬುಕ್ ಓದ್ತೀನಿ.
ಕೈ ತೋರಿಸ್ಬಿಡಿ, ಅವರು ಕಾಂಟ್ಯಾಕ್ಟ್ ಮಾಡ್ತೀನಿ. ಐ ಆಮ್ ಇನ್ ಟಚ್ ವಿತ್ ಹಿಮ್.
There must be many questions with students.
ಏನು ಇಲ್ಲ ನೋ ನ್ಯಾಚುರಲ್. ಒಂದೊಂದೇ ಮ್ಯಾಕ್ರೋ ಲೆನ್ಸ್ ತೋರಿಸಿನಲ್ಲ, ಸೊ ಅದು ಯೂಸ್ ಮಾಡ್ಕೊಂಡು ನೀವು ಒಳ್ಳೆ ಫೋಟೋ ತೆಗಿಬೋದು. ಅದರಲ್ಲೂ ತೆಗಿಬೋದು, ಬಟ್ ತುಂಬ ನಿಯರ್ ಹೋಗೋಕ್ಕಾಗಲ್ಲ, ನಿಮಗೆ ರಿಸ್ಟ್ರಿಕ್ಟ್ ಆಗತ್ತೆ. ಇದೆ ಮ್ಯಾಕ್ರೋ ಲೆನ್ಸ್ ಇದೆ. ಜಸ್ಟ್ ನೀವು ಅದ್ರ ಮೇಲೆ ಕ್ಲಿಕ್ ಮಾಡಿದರೆ ಆಯಿತು, ಅದು ಫ್ಲ್ಯಾಶ್ ವಿತ್ ಲೈಟು ಬರುತ್ತೆ. ಕ್ಲಿಪ್ ಬರುತ್ತೆ, ಕ್ಯಾಮ್ರಾ ಕಿಟ್ ಆಯಿತು. ಮೊಬೈಲ್ ಕ್ಯಾಮ್ರಾ ಇರುತ್ತಲ್ಲ ನಾರ್ಮಲ್, ಅದ್ರ ಮೇಲೆ ಕ್ಲಿಪ್ ಮಾಡಿದರೆ ಆಯಿತು. ನಿಮ್ಮದು ಯಾವ ಮೊಬೈಲು ಅಂತ ಹೇಳಿದರೆ ಅದಕ್ಕೆ ಬೇರೆ ಬೇರೆ ಇರುತ್ತೆ. ಸೊ ಅವರು ಆ ಮೊಬೈಲಿಗೆ ಹಂಗೆ ಅಡ್ಜಸ್ಟ್ ಮಾಡಿಕೊಡ್ತಾರೆ.
Thanks to Ishanya Sharma for his time. Unless and until you try practice, you cannot become photographer. Okay, so I'm looking forward to organize one workshop for students looking into this time. So probably in another next month or sometime, so we'll plan for that. Okay, so that for all the NSC students. So with the mobile or whatever, is it possible to take good photograph for thesis purpose? So, lastly, I will not take much time, maybe within 2-3 minutes I will finish off. So it will not be any valedictory function and I will not bore you. I will just thank whoever involved in helping me out in organizing this program and as well as the speakers. So first of all, I told morning that when I went, that Taisuke is coming, and I requested Vice Chancellor to support at least whatever possible. So he readily agreed that I will give the minimum amount, not for the flight charges of Taisuke. No, I told he is coming on his own. So he readily agreed and supported me. So that's how I could able to organize. So I thank Vice Chancellor sir and as well as all the patrons of the seminar like Hanumantappa sir, our Registrar Lokesh sir, Wali sir, Director of Research Chemla Naik sir, Dinesh Kumar sir. In fact, they extended all the facilities including Comptroller and DSW sir, our Dean Dushant Kumar sir and KC Shashidhar sir. In fact, Shashidhar sir also helped me now in with the financial support with his IDP project. And Hanuman Swami BC for allowing us to conduct the seminar in this hall because we wanted a medium-sized hall. So I thank Hanuman Swami sir and all the executive committee members and our seniors, S.U. Patil, Pradeep sir, the madam who was actually sitting since morning and now supported me in all along for conducting the seminar. Hanuman Rai sir who came from Modigere, Revana sir from Brahmavar, Vankarappa sir, yesterday he was there and now able to help me to arrange the things. Deshmukh sir, of course, is there throughout. Kenchareddy sir who came from Pannampet, Rashekharappa sir helped me a lot. Girish who came from Modigere and all the advisory committee members. Especially I thank Sushil sir and he gave the date. Okay, so that he could come and acted as chief guest and extended all the facilities and he also told that there will not be any problem for our students to go and work in NBAR. So that's how now I'll be thankful. I'll be incomplete if I miss the two of the foreigners who are there. I actually it will be live streamed in YouTube. Both John Sabatnik and from University of Life Science Prague and David were there. Okay, sometimes there were only two watching stream and those were from foreign. Okay, so I thank them and I wholeheartedly thank all the two veteran entomologists, especially who came from Manjana sir came from Asan and Vasanagada sir from Dharwad, who are actually the senior most, but they are the one who are actually noting down all the things. So Vice Chancellor was telling whether the students are actually noting down or not. So they were actually noting down and he had a keen interest in all the talks. Okay, so I am very much thankful to you, sir, and we request your support throughout. And all the students, of course, they extended our support so that bringing all the materials, arranging things, and registration and all. So, and I also thank Chikanna sir for providing good food in the lunchtime and also tea. So, whoever, if I have missed, I am sorry, but all of you really supported me in organizing this conference successfully. And Taisuke will be there for another 1 week and will be taking him for collection. Okay, thank you. Thank you, one and all.